The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO. Quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration, and each dream is driven by the passion to realize it. For over 50 years, We have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces, General Secretary and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration, and each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogoriense Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia-Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI also actions such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future. Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, eco-friendly transportation, advanced materials and nanotechnology research. Social sciences and humanities, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research. Scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. Studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogoriense Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia-Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders.
Wabarakatuh. Our plenary speakers, moderators, and all of distinguished participants. It's a great pleasure for me to welcome all of you on the second day of International Symposium on Applied Chemistry 2020. This day, we have a plenary session too, and also 2 parallel sessions at noon. But before we start this day, I want to tell all of you the several announcements to all of the participants and also our keynote speakers and moderators. First, please all of you have a stable internet connections. Make sure that computer has a working camera and audio device. In our symposium, the host will automatically mute the audio all of the participants, and the chat room is only available for delivering questions to the ongoing discussion in the session, requesting for changing or moving to the other breakout room in the parallel session, and announcement that delivered by organizing committees. Dear keynote speakers and also all of participants, don't forget to fulfill the list of attendees that we share the link in the chat rooms. And please properly rename your Zoom ID according to your participation category to set you up to the correct breakout room in the parallel sessions. For the participants that want to asking question, please state your name of institutions and also the panelist name that you want to deliver the questions. Okay, ladies and gentlemen, we will start the plenary session 2. In the plenary session, we have 3 speakers. First, Nilesh Prakash Nirmal, PhD, from Mahidol University, Thailand. Associate Professor Dr. Nurul Huda, from University Malaysia Sabah, Malaysia. Professor Nina Artanti, PhD, from the Center for Chemistry, LIPI. This plenary session will moderate by Dr. Renat Andri Frediansyah, a researcher from Research Unit for Natural Product Technology, LIPI. Dr. Renat Andri Frediansyah graduated from his PhD in pharmaceutics from University of Tübingen, German, during 2007 until 2020. During 2017 until 2020. His master's degree got from Chulalongkorn Graduate Institute of Thailand during 2013 until 2015. As a young scientist, Dr. Andri got many awards. First, ASEAN Youth Summit Fellowship in National University of Singapore, and also Best Student Research Award with the Japan Society for Environmental Chemistry, and also many awards. Well, ladies and gentlemen, I will give time to our moderators, Dr. Renat Andri Frediansyah, to conduct this plenary session.
Thank you, Dr. Marisa Angelina, for your very generous introduction. Ladies and gentlemen, welcome to the second plenary session of the 6th International Conference on Applied Chemistry, focusing on natural medicine and also nutraceutical. I am Andri and I will be your moderator for this plenary session. The session has 3 keynote speakers from 3 different countries, first Thailand, and then Malaysia, and our country, Indonesia. Before we begin, I would like to give information to all participants. At the end of 3 talks, we will have Q&A session for about 30 minutes. So if you have a question or something to be discussed, you could put your question in Zoom chat. Alternatively, you can press the raise hand button from the Zoom apps at the end of the 3 speakers. Okay, I think we're ready for the first speakers. The first speaker is food chemist from Thailand. He is a Dr. Nilesh Prakash Nirmal. He is going to deliver a talk entitled Biological Activities of Fish Protein Hydrolysate. Before Dr. Nirmal starts his talk, I would like to give an overview of his background. Dr. Nirmal is foreign expert at the Institute of Nutrition from Mahidol University, Salaya, Bangkok, Thailand. He achieved his PhD in food chemistry from Prince of Songkhla University, Thailand in 2011. He was also a team leader for Wild Bread in Brisbane, Australia back in 2018 to 2019. Dr. Nirmal also research fellow at the University of Queensland, Australia from 2015 up to 2018. His research interest is bioactive from plant and their derivative as alternative sources of food and food additive. He also interested in developing functional food product, food nanotechnology, seafood or agri-waste utilization, and also nanosciences. Dr. Nirmal also actively published his research in the top international journal, and currently he has a more than 35 journals. And ladies and gentlemen, please welcome Dr. Nilesh Prakash Nirmal. The time is yours and you have maximum 40 minutes to deliver your talk from now. Dr. Nirmal? Yes. Doctor, are you hear me?
Hello.
Yeah.
Hello.
The time is yours.
Audible? Am I audible?
Yes. Now, yes.
Yeah. Thank you. Thank you, Andrew, for your great introduction. And I would like to thanks the LIPI organizing committee for inviting me to this session. Can you see my presentation?
Not
Angie?
yet.
Yeah, can you
Not
see my
yet.
presentation? Yeah, okay.
Now yes. Yeah, now yes.
Okay.
You can start, yes.
Yeah. Yeah, well, I would like to thanks organizing committee for inviting me to give a talk on this occasion of 6th International Symposium on Applied Chemistry, focusing on natural medicine and nutraceutical. So today I'm going to talk on a biological activity of fish protein hydrolysis. So I choose this topic because we know all over the world there are a lot of people now who is moving from like animal proteins to the seafood proteins, uh, mostly like aquaculture or fisheries production. Okay, so the fish, they are moving towards more towards fish protein because of delicacy and easily digestible protein. So what we're gonna see in this topic today is the introduction, and then sources for fish protein hydrolysis, then production, purification and identification of any peptides, and their various biological activities. Further, we will see the structure activity relationship of fish protein hydrolysis, and additionally, we will see what are the challenges for large scale production and commercialization of this fish protein hydrolysis, and the research gap in nutraceutical applications. And finally, conclusion on this topic. So in introduction, as I told you that nowadays many peoples are moving towards the fish sources, seafood sources for the protein, like then these fish and fish products are well known because of their easily digestible protein, and the global fish production increases everywhere, including aquaculture and fisheries like the catching fish from seas, and the aquaculture like producing fish or other seafood in aquaculture like that. So we can see in table 1 the world fisheries and aquaculture production, which is produced and presented by the FAO in during 2020. From table we can see the aquaculture and world fishery production increases steadily from 2006 until 2018. We can see all the even sea capture or aquaculture production is keep on increasing because of demand from the consumers. They demand more and more like more is like use around 22 million ton is used for other sources like making feed product or animal feed product or other things. So in this, almost 27 of the total fish capture is unutilized because of loss of quality or storage problem or loss of quality. So somehow this 27% from this 170 million ton fish, they are utilized. That means they cannot sell in the market. So they have to either dump into the sea or they have to do find the alternative source for that. So, and then remaining harvested fish go through the processing plant, where only 20 to 50% portion of the fish is utilized as a edible product, and the remaining parts like 80 to 50% again discarded as a byproduct or leftover. So what this leftover or byproduct contain? This leftover or byproduct contain like skin, head, viscera, bones, scales, and fins. Because obviously we just like fillets and protein part. But how about this waste? This is completely waste. So some portion is used for the animal feed production, or some, if someone they don't have any technology or anything, they just dump in the land or sea anywhere, so it causes like environmental hazardness. So what we can use this either unutilized fish or the fish processing waste. So these byproduct actually contain 58% of protein and 90% of fat and some content of minerals. So it has the potential to become reutilized. So we can reuse this waste, we can make some process, and yeah, we can utilize either for, again, for animal feed production or for human consumption again. So what we plan here, like these uh, these waste or waste product can be utilized to produce the fish protein hydrolyzate. So fish protein hydrolyzate, so far there are a lot of research that it shows the fish protein hydrolyzate containing active peptide exhibits various biological activities. So now we will see what are the sources. So as I told you, sources, it can be fish flesh or muscle, like we usually eat directly, but we can use that flesh or muscle to produce a fish protein hydrolyzate also. The second is fish skin, and then other byproduct, which include like scale, viscera, head, and fins. So fish muscle or fish flesh contain the myofibril protein or sarcoplasmic protein. Then fish skin is high in collagen. Similarly, these scale and head, they also have other proteins, including collagen and other protein sources. So we can use these protein to produce a fish protein hydrolyzate. So for this, what are the production technology? How we can produce the fish protein hydrolyzate from all these raw materials? So raw materials, it can be either whatever available for you, or you want to utilize the waste, or you want to make from flesh or anything. So there are 4 different methods so far, like people are using mostly for the production of fish protein hydrolyzate. The first, enzymatic hydrolysis. Enzymatic hydrolysis. So enzymatic hydrolysis contain, obviously as the name shows, enzymatic. That means they use enzymes for degradation of proteins. So enzymatic, so there are different kinds of enzymes like endopeptidase, exopeptidase, and all that. And now we can get in the market like alkalase, Protamax, Nutrase. And there are a lot of varieties of proteins, commercial enzymes available, which you can use in the process of enzymatic hydrolysis. The second is chemical hydrolysis. Chemical hydrolysis, the very first, they use like acid hydrolysis or alkaline hydrolysis, but these are like really hazardous to operators and environmental also. So chemical hydrolysis, you can use either alkali or acids. And the third is auto hydrolysis. So what is auto hydrolysis? Auto hydrolysis is using the enzyme which are already present with the raw material. Every organism or everything, they have a proteolytic enzyme. They just give the active condition, including pH and temperature. So these enzymes get activated, and these proteolytic enzymes, they degraded the protein itself in the same part. And the fourth one is a fermentation hydrolysis. So fermentation, as it says, fermentation. So fermentation, they use a different bacteria to ferment the medium, and the medium contains either your fishery waste or any, like whatever you want to get the hydrolyzate. So there is like again, you have to get the active pH and some mineral contents of the media, so that the bacteria can grow and produce. Again, there is a proteolytic enzyme or a protease enzyme, variety of protease enzyme depend on the bacteria source you use. They produce all these enzymes, and when they produce the enzyme, these enzymes cleave the raw material, which is like very now complex, but they utilize, as their growth time increase, they cleave, and they produce the small fragment like peptides from the proteins. And then, yeah, so this is the four way that we can produce the hydrolyzate from protein, large molecule protein to a hydrolyzate. So what they generally do is we collect the fish source and then we homogenize raw material with the water, adjust the pH and temperature as per the requirement. If you are going to use like enzymes from external or autolysis or fermentation, so we need these pH and temperature active so that these enzymes can be active and it can completely degrade the protein. Then addition of enzymes and hydrolysis. Then we have to add the enzymes, and then there is a particular time depend on the procedure and everything. We have to allow to completely hydrolyze the protein into the hydrolysate. Once you make sure that now the hydrolysis is done, then we can deactivate enzyme. We can deactivate enzyme by changing the temperature or by changing the pH. Once the deactivation is done, centrifuge hold the content and then collect the fractions, collect the filtrate, and that filtrate is your fish protein hydrolysate. Actually, it's a protein hydrolysate, but because we use the fish sources, so we call it as a fish protein hydrolysate. So what is the next step after this? The next step contains the purification and identification. So now it is depend on your research interest that whether you want to go for purified or you just want to use like a crude, whatever you prepare fish protein hydrolysate. But now, because due to the a lot of competitions, many researchers, they wanted to purify it, they wanted to identify what exactly the peptide sequence which are giving a particular biological activity. So that is the interest of scientists. So they, for that, they go for the purification. So purification obviously follows the chromatographic techniques, which control filtration, ultrafiltration, gel filtration, size exclusion, affinity. And there is one more I would like to add, like any enzyme bioactivity guided assay. Okay, so we can use that technique also to purify your peptide. So all these chromatographic techniques like size exclusion, which can exclude the smaller size first and retain the large molecule. So this is what affinity, like based on the positive and negatives. And wherever you get the highest antioxidant activity, we use that fraction for further identification or further high purification. So for that, we go with the identification, go with the LCMS and Malditov. LCMS is the liquid chromatography coupled with mass spectrometry. Malditov is the metric assisted laser deposition, ionization, time of flight technique. So these techniques are work on the like ionization of the molecules, and then they detect that molecule and molecular weight and everything. So here you can get actually the molecular weight, also the peptides, and the sequence also you can get. There are another techniques like peptide sequence and all, you can do that also. So the next step is the biological activity. As I told you, this fish protein hydrolysate. So they shows the various biological activities. So far, a lot of biological activities have been screened, and there are still new biological activities going to be screened by researchers all around the world. So biological activity of the proteins are influenced by the primary structure of protein stability, amino acid composition, and content of that amino acids in the peptide sequence. Additionally, the bioactivity of protein hydrolysis depend on various conditions used during the hydrolysis. As I talk about the production, we can see that during the hydrolysis, like the types of enzyme, as I told you earlier, exopeptidase, endopeptidase, or the enzyme to the substrate ratio, pH, temperature, and time of hydrolysis. All these factors define the final product or the final product composition of your hydrolyzate. Whether it can be a small peptides or it can be a large peptides is almost depend on your time of hydrolysis and the purification technique you use and everything. So once you get this fish protein hydrolyzer, this fish protein exhibit for biological activity screening. So far, they have reported like majority of the activities, including antioxidant, antimicrobial, antihypertensive, calcium binding ability, anti-diabetic, immunomodulation, and anti-proliferation activity or anti-cancers activity. So this is what the overall, what fish protein hydrolyzate can give to our body, or how it can be a benefit, or how it can, why it can be used as a nutraceutical applications. There are a lot of like some miscellaneous activities also with this, we will see. Now, antioxidant activity. So these days, many, almost everyone now, they want some antioxidant. Wherever they are just looking on, oh, I'm looking for food which has high antioxidant, or we are screening for the phenolic compound from plant source or this source, which should have high antioxidants and microbial and other activity. So this antioxidant activity is very important actually, very, very important, because in normal living being, we actually the oxidation happen in every part of cells, or even in human body or plant also. Oxidation is the normal process of the life cycle. But sometimes the over oxidation can create a deadly or notorious reactive oxygen species, which is such as like superoxide anion radicals, hydroxide radical, hydrogen peroxide, and singlet oxygen. So once these excess free radical form, they are really unstable and very reactive. They can damage your cell protein, including nucleic acids, even lipid. They cause very serious damage to the cell and subsequently the human body. So that's why we need antioxidant. We need action antioxidant. That's why many people, they are looking for a food like keep healthy lifestyle and trying to get the food which can give you like antioxidants. So we mostly they go for like a fruits which produce the most phytochemicals, which are known antioxidant, highly antioxidant. So another things I would like to tell you, how these ROS or reactive oxygen species, they form actually usually as a oxidation process. But the one important factor is stress, which increase all these things, which increase the number of reactive oxygen phase and inflammation, and it act like a precursor for all these things. Yeah, so now what these antioxidants do, they are the stable active chemical compounds. They are capable to intercept, combat, and prevent or reduce the reactive oxygen species. So how these antioxidant molecule they do? They do by different process. So usually there are three mechanisms, like scavenging free radicals, like these free radicals, or quenching the singlet through metal ion in through phantom reactions, and they increase the uh, oxide superoxide radicals. So that increase like reactive oxygen species. So far, there are different analytical and cellular method used to assess the antioxidant capacity of fish protein hydrolyzate. So what are the analytical methods? The analytical methods consist of like free radical scavenging activity like DPPH, superhydroxide, then hydroxyl radical scavenging activity, then metal scavenging activity, then ORAC. ORAC is the well-known analytical method. Plus, there is like lipid peroxidation inhibition activity. These are the analytical activities which you can do in the class on a chemical basis. And there are some cellular. So nowadays, many people, they prefer cellular antioxidant because it resembles more to human body or cellular activity. So cellular antioxidant activity consists of like antioxidant activity in cells. Then lipid peroxidation inhibition in human red blood cell. Another one is inhibition of oxidative DNA damage or protein damage in the cells. So these activity you can conduct through cellular methods. So in that, the antioxidant mechanism of fish protein hydrolyzed largely depend on the amino acid sequence of the peptide. Now, so what is the, excuse me, the antioxidant mechanism of whatever you have hydrolyzed or purified compound is largely depend on the amino acid sequence. Yeah, obviously everyone knows protein is the long chain or amino sequence of amino acid. Protein consists of various amino acids and then it is formed as a protein. So these amino acid also has the side chain active molecules which are involved in various activity. So the most reactive amino acids are like acidic side chain, aspartate and glutamate, and sulfur containing side chains including cysteine and methionine, and aromatic side chains. Additionally, shorter peptide, shorter that means low molecular weight peptide generally show higher antioxidant capacity compared to larger peptide or polypeptide. This is obvious because small molecular weight peptide are easy for access to the reactive oxygen molecules, or it can easily penetrate within the cells and it can easily react. However, the large polypeptide, because of large change, it has some adverse reaction here and there, and the active molecule is not available to counteract or interact with the radical ions. And you know, the aromatic side chains, they are well known metal chelating because of phenolic ring. So they have ability to chelate with the metal ions. Now, the next one is the antimicrobial activity. So why the antimicrobial activity also? Yesterday we have seen Professor Chaudhary. He have mentioned about the multi-drug resistance bugs. So we know how these bugs, they are deadly bugs. So researchers all around the fields or everyone, they are trying to find a natural source, including plant, herbs and everything, getting phenolic compound to try to find some bioactive compound which can kill these microorganisms which are really deadly to human beings. So there, okay, how these act, because these hydrophobic amino acid residue, they are actively interact with the negative charge surface and phospholipid membrane of the bacteria, result in disturbing cell wall and killing them. As we know, I hope everyone knows the bacterial cell wall and everything, it has the negative charge and phospholipid membrane. So mostly these small protein hydrolyzed or small peptides, they are able to interact with this negative charge, and they are able to, after interaction, they can disrupt the cell wall and then cell lysis. This is how they kill these bad bugs. That's why this is a very important activity which can be shown by fish protein hydrolyzation. Another one is angiotensin 1 converting enzyme. So now hypertension. You know, hypertension is the leading cause of heart disease. So why angiotensin 1 converting enzyme? What is the role of this enzyme? So what this enzyme do, first I would like to tell you, this is a non-specific dipeptidyl carboxypeptide present in renin-angiotensin system. So what this renin-angiotensin system do? This system regulate arterial blood pressure and fluid hemostasis. So it regulate the blood flow. Okay. And ACE is responsible for increasing blood pressure by converting angiotensin 1 into angiotensin 2, which is a vasoconstrictor. Okay, and also a deactivated of protein vasodilatator. So like this, it choke your veins and everything, and which cause the hypertension. So therefore, to inhibit this ACE enzyme is commonly used as a first line of therapeutic treatment to control the hypertension. So this protein hydrolyzate, which can shows the ACE inhibitory activity, they are mostly, these inhibitory activity depend on amino acid composition and the molecular weight of the peptide. Okay, so the presence of hydrophobic amino acid, again here hydrophobic amino acid, arginine and valine at the C terminal. So please note that what we have found, most of the biological activities of peptides, they show the activity, and these peptides contain either hydrophobic or any particular amino acid at the C terminal, carbon terminal, which increase the ACE inhibitory potential of the peptide. There are some in vivo study as well. So it is not just in vitro, but there are some in vivo study uh, where fish protein hydrolyzate given to the spontaneously hypertensive rat. The content was like 600 milligram per kg body weight, which shows significant lower systolic and diastolic blood pressure. So it is not only the flash analysis or analytical process that it can inhibit the AC inhibitor activity, but there are some mice study or rat study which shows the successfully they can lower the hypertension in hypertensive rat. Another activity is calcium binding activity. Now why this calcium binding activity here? Okay, as we know, calcium is an essential element for all living organisms. Calcium play a vital role in many cellular mechanisms. Even they are helpful for bone formation and then everything. So this is really important for uh, well function of the body. So calcium deficiency is the worldwide problem owing to insufficient intake or decreasing the solubility. So actually, this is the major problem. Usually after age of 30, our calcium is, our body is not able to absorb the calcium from whatever the food we have eaten or whatever the tablet we eat. Our body is not absorb that completely calcium. So there is a problem for the solubility of calcium or availability, bioavailability of calcium in the body. So that's how this fish protein hydrolyzate play a role here. Calcium ion bind with amino nitrogen and carbonyl oxygen atom of fish protein hydrolyzate. So once this calcium, like the major amino acid responsible for strongest calcium binding properties of peptides are aspartate and glutamate residues. So when we have this complex, this fish protein hydrolyzate and calcium complex, it has the great solubility, and it's increased the bioavailability for our body. It not natural protein hydrolyzate from fish waste or uh, any fish sources. So this DPP4 inactivate incretin, which is involved in insulin secretion. So reduced insulin secretion cause type 2 diabetes. Therefore, this is very important to control this enzyme. So how? So these peptide fish protein hydrolyzate, which can inhibit this dipeptidyl peptidase 4 activity, it contain amino acid, mostly proline, glycine, or hypersin. These amino acids are mostly found in collagen and gelatin. So then fish protein hydrolyzate, it may protect the total circulating incretins, activate GLP secretion, decrease plasma glucose, and also improve glucose tolerance by inhibiting inhibiting DPP4 activity. So most of the researchers, they not only just study analytical methods, but they did some cellular or animal study also, using mice, or some studies include human being also, clinical trial. The next one is immunomodulation activity. Immunomodulation. We know the immune system. Immune system is the most important for well-being, right? Immune system is influenced by many internal and external factors such as stress, unhealthy lifestyle, pathogens, and foreign antigens. So to modulating immune action is must for health and immunity of the body. So how to modulate this? So actually there are a lot of ways, like exercise, eating habit, many other things which can boost the immune activity. The immune response triggered by bioactive peptides considered to be one of the form of immunoregulation. So here it is like bioactive peptide, how they boost this immunoregulation. So what these peptides do, they upregulate the immune function by different route. Upregulation of immune activity, such as increase of TGF. TGF is the transforming growth factor beta. They increase the transforming growth factor beta, and interleukin 10, also splenic NK, natural killer cell toxicity, and macrophage phagocytosis, and level of serum immunoglobulin A. So they increase all these activities of all these uh, cells and the activity of molecules, so that immune system get boosted up. By doing this side by side, they also down regulate the pro-inflammatory cytokines, including TNF alpha, that means tumor necrosis factor alpha, then interleukin 6 and 1 beta, then nitric oxide, PGE2, prostaglandin E2 production by deactivating NO synthase and COX mechanism. So far we saw all these fish protein hydrolyzate or peptide, they are having activity. In each activity, their mechanism of action is different for every biological activity. The next one is anti-cancers or proliferative activity. Yesterday we have seen our keynote speaker, Dr. Ambara, right? Yeah, Dr. Ambara, he have talk about the breast cancer. He have talk about the breast cancer, and he have shown like how this synthetic probe can help to recover the cancer patient, and how it can reduce the anti-cancer activity and all that thing. So the same, like as I already told you, there are different sources which can work differently in every reaction. So what this cancer is defined as abnormal growth and proliferation of cell in the body. Therefore, controlling the initiation or promotion of cell proliferation is an important task for treating any malignant tumor. So this is the major step, like how to control. We have to control this malignation or initiation or promotion of cell at the start itself. So here, these peptides, they work very well. They shows this anti-proliferative activity through cell toxicity, cell cytotoxicity, and other mechanism like apoptosis and blockage of nutrient. So you can see this is a natural, produced from a natural source, and a very like body friendly, you know. It may not have any more inflammation or something to your body, because it is prepared from natural source, and it can work through apoptosis and blockage of nutrients. So yes, it is a promising future research for this fish protein hydrolyzed. Now, so these are the mostly known activity. Now, other reported activities. So there are other reports, people, as I told you before, researchers, scientists, they are still looking as these peptides, they are has a versatile biological activity. What else they can do? This is the eagerness of scientists, what else they can do. So they try for different. So far, antithrombic activity, anticoagulant, then anti-obesity effect, anti-fatigue, anti-atherosclerosis, anti-aging, anti-glycation, cardioprotective, neuroprotective, hypocholesturemic, and wound healing activity. Yes, all these activities, this fish protein hydrolyzate has a capacity or has a tendency to act in all these disease condition or to improve the health of human being. Now, so what importance, how all this fish protein hydrolyzate shows this biological activity. Important is structure activity relationship of fish protein hydrolyzate. As I told you that the most important thing, the structural characteristic of fish protein hydrolyzase, which includes molecular size, chain length, amino acid composition, and sequence, and their functional group. These are the most important thing, because these things, like mostly the amino acids and their functional group, they are involved in the all these biological activities. And obviously the size, because smaller size, it facilitate more interactions, it increase the interaction with particular area or disease or uh, reactive species, such as like this structural activity of fish protein hydrolyzase is different for different biological activity. The most bioactive peptide consists of hydrophobic amino acid in peptide sequence, basically at C terminal, carboxy terminal. So this is what they have concluded, that the most active, it means it can show various biological activity. This protein hydrolyzate has hydrophobic amino acid at the C terminal. Nevertheless, molecular size, chain length, and functional side chain of amino acid also influence the structural activity for the particular bioactivity. Now, after we seen that this fish protein hydrolyzase is so much promising, it's like is a really good medicine for most of the disease. It has ability to cover, and it has ability to improve um, many things. So what is the problem then? Why there is not many products in the market? Actually, we have some product. There are some product, but those products are like fish protein collagen hydrolyzate, but in a particular range. It is not any single peptide or anything. So what are the challenges? Challenges are the continuous availability of sufficient raw material. As we know, yes, this is the very important. Is quite different from the lab preparation. Reproducibility and consistency in batch to batch production, that is also another challenge. And the next one is the stability of bioactive peptide during production and storage. This is the biggest one. And the third, for commercialization, cost of the raw material, production process, and marketing. So these are the things while they go for the large scale production and commercialization. Next, nutraceutical application. Still, as I told you, there are very few products, that too from the Japan. They are selling some gelatin hydrolyzate or protein hydrolyzate, but they have something like only like 2000 kd up peptides or something like that. It is not any isolated peptide or something. So what is the nutraceutical research gap is the toxicological study of particular peptide sequence. Then pharmacokinetics, the one we have to know before any clinical application or nutraceutical application. We have to study all this toxicological study, pharmacokinetic study, and then long-term effect of consumption. We have to study that also. So if we consume every day, like long, like 6 months, 1 year, what will be the effect on our body? Because we will taking this as a supplement or nutraceutical added. So what is the effect? Then large number of clinical trial for particular disease condition. The next, safety, environmental and legal consideration of raw material and final product. Yeah, legal consideration is very important. That's how the regulatory harmonization all around the world is needed to become acceptable nutraceutical product all around the world. The conclusion. So this is the wonderful area and growing area. Due to its various biological activities, researchers are still interested. They are still utilizing new fish sources, various different parts using different processes. They're trying to get the different easy, fast, cheap processes to get the higher production and many things. There are a lot of scope for this. And then sources like unutilized fish, like there are some small fish sometimes. When fishers, they go for the catch of fish, they get the fish which is people like we don't eat, so they just throw it or dump it. But they can use these unutilized fish, then they can get the fish muscle, fish skin and other byproduct. This protein hydrolyzers show different in vitro and in vivo biological activity. The gap between commercial production and nutraceutical application of fish protein hydrolyzers should be overcome. The biological activity of fish protein hydrolyzers open the door for nutraceutical industry. These are the references, actually just small references I'm showing here. There are a lot of references, and moreover, you can get from our publications also. There are a lot of, you can just search my name. I work on shrimp processing waste also. So a lot of different and various application and waste utilization, which is published in Trends in Food Science and Technology this year. Yeah. So thank you very much everyone for your attention and listening me. However,
Okay.
yeah, Andrew, I would like to request you, if I have another 5 minutes or 10 minutes, I would like to promote my startup, my business here. Can you allow me to do that?
We still have around 6 minutes for you. Maybe you can start
6 minutes.
from now.
Wonderful. Yeah. Thank you for allowing me to show my business. So I'm happy to tell everyone and announce that I have started the business with Hillberry Life Sciences. This Hillberry Life Science is the production, everything is in India. And what we are doing, we are a manufacturer of herbal extract, essential oil, and dietary supplement, capsule formulation. So all the materials are Indian origin, and we have the state of art of manufacturing facility with GMP, halal, FDA certification, then ISO, HACCP, GMP certification and everything. So as per your need, we can make all the supplement, including immune booster, then for bone osteoporosis and a lot. So what our services offer, we offer the best manufacturing services. We offer a contract research and development services and contract manufacturing. We do have our products in the market, like you can see, like Blackmore and all those, the nature products. They have their own product for each particular disease. So we also have that. And not only that, we can produce capsules or do the stability study or regulatory study for any product which you would like. So if you are interested for any quotes and order, please contact me on this email, nirmalnp21@gmail.com. And here you can see my mobile number. This is the Thailand code 66 and my mobile number 8345 72612. So here I would like to offer one more chance for those who are interested to do a business. Those who is interested to do business with us, I'm welcoming them. Please contact me. The business, like, you have to get the industry contact from your country or all around the world, and let's start the business, commission based business. So you can, this is a really good opportunity. If anyone interested, please contact me. And here is my email address and my mobile number. If you not get, then you can call, contact the Abdi, and you can get my contact from him. Thank you very much. Thank you very much. And thank you, Andre, and thank you, organizing committee, for having me here. Thank you.
Thank you. Thank you, Dr. Nirmal, for your very great talk. Your talk about biological activity of fish protein hydroxylate for human health. That's very useful for everyone. And also good luck for your startup. I hope everyone could join with you.
Yes, yes.
If you
Thank
have
you
any
very much.
question later, if you have any question for the audience, you can ask in the chat and we will discuss in the end. And
Sure.
because the bad connection in Malaysia, so we wait for Professor Huda until he could join with us in Zoom meeting. Therefore, we're going for the next keynote speaker from Indonesia.
Okay.
The speaker of this plenary session is from Indonesia. Yeah, I told you. Are you ready from Professor Nina Artanti? Are you there? Just to make sure first.
Yep. Okay.
Yes. Okay.
You can hear me?
Yes. Okay. She is a professor, Dr. Nina Artanti, and she will deliver her talk about Indonesian biodiversity of source of functional food. Before Professor Artanti starts her speech, I would like to introduce her professional background. Presently, she is a research professor at Research Center for Chemistry, the Indonesian Institute of Sciences, or LIPI, Indonesia. She achieved her doctoral degree from Ehime University, Japan, in Applied Bioscience and Biotechnology. In 2007, she was also fellow holder for a short course conducted by JICA Bioindustry Group Training in Japan. She has also received a 2-month research fellow from participating in training on antioxidant bioassay and hepatoprotective at Zhejiang University, Guangzhou, China, with Professor Pan Yuangjiang and also Professor Lu Jia as a part of LIPI Zhejiang University in traditional medicine collaboration. Currently, her research interest is broadening from the biological activity of natural product and also functional food. Professor Artanti actively published her findings as a part of dissemination in more than 20 papers in Scopus, and she was also appointed as a reviewer in various top journals. And ladies and gentlemen, please welcome Professor Dr. Nina Artanti. The time is yours and you have about 40 minutes to deliver your talk.
Thank you very much, moderator. Okay. So the share screen is okay?
Yeah, we can see your slide now.
Thank you very much for this opportunity. Assalamualaikum warahmatullahi wabarakatuh. Today I will give a talk on Indonesian biodiversity as a source of functional.
Professor.
Yes.
Could you increase your voice?
Oh, yeah, that's right.
Volume, please.
It's okay.
Yeah. Yes.
Okay. All right. I will deliver my talk about Indonesian biodiversity as the source of functional food. Okay. Indonesia is a large country. Its land is 1.9 million kilometers square, and we have 17,000 islands on when only 6,000 is inhabited, And for the sea territorial is also large. So then the population is about 274 million people. As this large country, we also known as one of the world mega biodiversity country. Diversity comprised of 1,500 species of algae, 80,000 fungal species, 595 species of lichen, 2,197 fern species, and many flora, accounting of 15.5% of the world flora. And in terms of terrestrial, we are probably number two after Brazil, but we also have the marine. That's why we have maybe the biggest biodiversity stores. And we usually already know about this biodiversity will be used for to find new drugs and everything. But of course, it's also a good source for functional food. Is it okay or because
Sorry for your
okay?
very sorry
Oh,
for
okay.
Yeah,
All
yeah,
right.
true.
I
No,
thought
no,
that
it's
is a close connection. Okay. And so, what is I also think that this biodiversity should be also explored and used for the development of functional food. And what is the functional food? There are many definitions about functional food, but they all uh, correlate to each other. That is a food that will be more than just us to have the nutrition, but also to uh, have effect on health uh, and should be consumed as a food. So it's probably differ from the nutraceutical of something that then can consume as a tablet or something. It should be good and have a taste nice and so but so it will be a bit different from the development of the uh, bioactive compound as we use it for drugs or herbal. So this many definition in here according to Academy Nutrition of Dietetics, International Food Information Council, Institute of Food Technology, International Life Science Institute, European Commission, Health Canada, and the Japanese is known that for in the history that start calling the functional food. Unfortunately, in Indonesia, the term of functional food is not in the Badan POM, the Indonesian Drug and Administration, Drug and Food Administration Association. What do they call it? Sorry, and because they call it before it's in 2013, it's the term of functional food is there, but then they move it. They simply don't want to have it anymore and just call for food with special health claim, and it's so only the one that already have a good uh, probably clinical studies things that can be used that claim for that functional food. So there is about 15 ingredients that in the world approved as functional food compound, which is a vitamin, mineral, sugar, alcohol, unsaturated fatty acid, peptide, and protein, amino acid, dietary fiber, prebiotic, and probiotic, and so on. And it's also have to be a permitted health claim uh, because it's already a lot of clinical studies and clinical studies. So it's like calcium and will relate to osteoporosis, dietary fat, cancer, dietary saturated fat, and cholesterol related to coronary heart disease, and fiber containing grain products, fruit, vegetables, and it's good for probably reduce the cancer risk, and fruit and vegetable grain products that contain fiber, particularly the will be reduce the risk of coronary heart disease and sodium and hypertension. Fruits and vegetables cancer, uh, of the digestive system. So this is the very common uh, food and ingredients that we could found in many of our biodiversity, and we should be used to explore it. So this
Professor,
also,
sorry for interruption again.
yeah, yeah. What
Uh,
the
could
sound
you
is?
increase again your voice? Yes, because
I
I
see.
got chat.
It's all the same. Sorry. Okay, there's another
Okay,
is a folate,
thank you.
soluble ISAC, sugar, alcohol. Do not increase dental caries, soy protein to risk for coronary heart disease, and phytosterol and phytosanol in the coronary heart disease addition, whey protein, and also the green tea extract related to reduce the coronary heart disease. And very well, like arrowroot, gembili, gembolo, suwak, porang, iles-iles, uwi, taro, pers, and kana. Because in major agriculture, only usually just use cassava and sweet potato and also potato now. But the other is just less used. So it's actually a good source of uh, starch like inulin and also anthocyanin, glucomannan, which has low glycemic index, which is good for diabetic people. So we should use we explore the other tubers in Indonesia to use it as a source of functional food. I think our colleagues in Gunung Kidul and also in Subang also try to make the functional food based on this underutilized tubers. Maybe if I can go back, sorry, this is the ones that suwak is the same family of the bunga bangkai, and this is I think the black potatoes, and I also there's some is alas this one, and in biotech before we already tried to make the propagation with the tissue culture, and this the cassava, but we increase the beta carotene to get the more beta carotene to make it yellow, so it has a higher beta carotene content. I forgot this one. What's this? Okay. Uh, so this is also some underutilized vegetables. This is kucai, and there's this pegagan, in this kelor. I think the this labu, I don't know the English name, is more still not to utilize, and this something like grass that's rarely used. But this is some medicinal traditional of some Indonesian underutilized vegetable that's have the medicinal or traditional use. So it can be developed into making the functional food, like for the allium, it's for antihypertensive, and also the centella that we know that have many activity. That's I think is also known as a part of cosmetics, used for cosmetic, beside of the good in also for memory enhancements. And daun moringa is calor is also good for the content of protein, and besides it's good for antihypertensive, diabetes, and also heart disease. This underutilized vegetable will rich source of polyphenol and ascorbic acid and carotenoid. Mm. And we in Indonesia we eat a lot of rice, and most of us I think usually just eat the white rice. But then actually there is many variety of rice in Indonesia, and like the black rice, and some is a red rice. And now because the people more aware about the health, so a lot of people now start eating the red rice. So there is some variety of black rice here that it will be published that may be a good source of because it have a good low glycemic index compared to the white rice that's supposed to be good for diabetic people. And this is have a functional bioactivity studies conducted in antioxidant, anti-hyperlipidemic priority research that we got the functional food flagship is one of the uh, commodity that uh, we should use to develop the functional food. As the largest, Indonesia is the largest center of banana diversity in the world. More than 200 local cultivar and at least 15 wild variety of Musa acuminata. Banana belongs to the family of Musaceae that's consisting of 70 species. Then genomically, banana is divided into three genome AA, BB, and SS. Banana fruit is useful for is alternative of staple food raw material of processed food with high carbohydrate and potassium that had potential for non-communicative to 138.5 nanogram per milligram for alpha cryptoxanthin and 3.9 to 29.4 nanogram per milligram for beta cryptoxanthin and 3.5 to 80 for alpha carotene, 10.8 to 118 for beta carotene. These results show that 4-carotenoid content was very small as compared to total carotenoid content. This suggests that those 4-carotenoids were not a major component of uh, Pandanus conioides carotenoid. Using the principal component analysis, 9 clones of Pandanus conioideus can be grouped based on the proximity of its carotenoid content into group A, group B, and group C. This is where they're found. This is the called the buah merah, and it's also in Papua. Is traditionally used for the uh, oil of that, so and also for food. So it's very good for try to develop as a source of functional food using this plant. And we found also that Indonesian rich on exotic fruit, and there is a publication that uh, this they want to use the exotic fruit seeds to to use it as functional food because they are rich in lipid and also fiber, protein, and not as carbohydrate that probably useful to develop into the functional food. So they contain many nutraceuticals like polyphenols and also flavonoids and tannins and phenolic acid such as quercetin and as caffeic acid. And besides, Indonesia also rich in animals with potential bioactivities. Pangolin is often smuggled to countries like China. Apparently, the pangolin is the potential to be raw material with omega-3 content in EPA and DHA that reduce cancer rates, reduce inflammation, hypertension, arthritis, and maintain brain function. But I think because of the COVID-19 pandemic, or people will be uh, don't want to use the exotic animals like this to be eating because afraid of the possibility that they get generated, even though we are not sure about it. That the scales of Mandarin also contain tramadol, HCl, an analgesic, an active substance for the pain relief, as well as belief can cure fever, skin disease, and venereal disease. Eating meat we believe to be efficacious of aphrodisiac. Extract of roxin contain of alkaloids that can be used instead of morphine for pain relief. In addition, frog skin extraction also contains a substance that is able to active the pancreas gland that can be used as an anti-diabetic drug to stimulate insulin. There are at least 347 species snakes in Indonesia. Traditionally, people use the extract of whaling snake at Bungarus candidus to lower blood pressure. Like the diversity of flowering plants, Indonesia also has a wealth of bees. Traditionally, bee venom may be used for rheumatic therapy, arthritis, lumbago, and skin disease. The bee venom contains apamine, melittin, phospholipase, hyaluronidase that suppress the nervous system, stimulate the liver, and adrenaline glands. And for the marine, from the marine, we have a lot of sea cucumber species that have a lot of bioactive compound that is long been used as food or traditional in Asian countries, and the price is actually expensive. And actually, there is a 350 sea cucumber species from Indonesian water have been recorded, and at least 26 with economic value. And like Stichopus hermani, Talentota ananas, Terentota anax, and Holoturia fugolia, and Actinopiga mauritiana as most highly valued species. These organisms are potentially source of high value added compounds with therapeutic properties such as triterpene glycoside, carotenoids, bioactive peptides, vitamins, minerals, fatty acids, collagen, gelatin, chondroitin sulfate, and amino acids. In the recent years, health benefit effects of sea cucumber have been validated through scientific research and have shown medical values such as wound healing, neuroprotective, anti-tumor, anticoagulant, antimicrobial, and antioxidant. This functional material lead to potential development of various food and biomedicine industries. And Indonesia also known with a lot of fermented food, and we know there's a lot of fermented food is good for the functional food because they contain of lots certain acids that's good for our body. And this is some of the Indonesian fermented food that and the micro organisms that make the fermented food. Because like tempe is the Rhizopus oligoporus and Rhizopus aureae from Paris bean, but the most common one is soybean. Although it also can make from other beans. And ketchup also size. The sweet ketchup I think is traditionally of Indonesian because the other Asian countries mostly like the only the salty soy sauce. And also tauco is also based on kedelai, and for the sweet sour sauce is based on kedelai, but soybean, I mean, and but for the uh, salty soy sauce is usually sometimes use the fish. And oncom is also from soybean, but some is from peanut. And tapai is from singkong or cassava, and rice with fermented with Saccharomyces cerevisiae or yeast usually. But also the because it's not a pure culture, it's sometimes mixed with amylose, rossi, and Rhizopus chinensis. And daddy is a fermented milk that's have lactobacillus, so the lactic acid bacteria, and also brum. That's also from using fermented using yeast, and tuak, that's also from nira, uh, coconut, and aren, from fermented using the Saccharomyces cerevisiae. And nata is now the common that we from the uh, coconut water, and it's using Acinetobacter cereum. The red oncom is fermented of solid water of tofu by Nerosporas storophylla, and isolated from oncom has been studied for carotenoid production as an alternative for food and beverage colorant. Black oncom, produced by Rhizopus oligoporus, with raw material of peanut and peanut soybean cake mixed with ongok or tapioca flour waste. Active compounds are isoflavone, genistein, daidzein. And angkak is fermentation product of rice with monascus, used for natural food coloring, species, and medicine by Indonesian people who believe in pharmacological property. Monacollica has been used in clinical therapy to lower blood cholesterol concentration. But as a functional food, it can be used as the part of the food. But if we only use it for the active compound, maybe it can be powder and then like a nutraceutical put in a capsule or something like that. And from the microorganism and in Indonesia, we found the nonomuria. It converted inulin into the DFA3. It's with the enzyme inulin fructotransferase. This from the 100 isolates of actinomycetes from soil sample collected from the province of East Kalimantan and West Nusa Tenggara screening for inulin fructotransferase. The supernatant of strain ID 06A0189 is find to be converted inulin into a compound having an RF similar to the reference standard of fructofuranose, with the anhydride, we call it the DFA3. This strain is identified as Nonomuria sp. In the it belongs to the Streptosporagiaceae family. This is the first report of inulin fructotransferase produced by this family. The DFA3 has many characteristics that are beneficial for functional food processing. It's a non-reducing sugar that is soluble in water, has half of sweetness of sucrose, is stable in acidic condition at high temperature, is resistant against the browning reaction and the non-caloric sweetener. The FH3 has several health benefits, enhancing the uptake of calcium, zinc, and magnesium in the small and large intestine of rats and humans, inhibits the hydroxy reaction of bile acid to provide secondary form, which is a risk factor for colon cancer. Therefore, the FA3 is expected to be a new prebiotic candidate. So is the now I will talk about the some that's research in the of functional food in Research Center for Chemistry. This is a natural folic acid. It's work from Miss Agustin that I think some of you already heard her presentation yesterday, and there's another one today related to these studies. So we make the natural folic acid based on the like tempeh from soybean and also from uh, green bean and from the uh, nixtamal of corn, and as well as some fermented vegetable using lactic acid bacteria, and then we make it as a source of natural folic acid by identifying using the LCMS to make sure that we got the certain amount of folic acid and use as a food to make cookies for babies, but we just try to make it, and maybe we should collaborate with our colleague in Subang and Gunung Kidul that's already have experience in collaboration with the Puskesmas, and also to make the possible for the clinical studies. So this can be uh, told or collaborate with small medium price enterprises or with the industry, food industry, that will be interesting to use this product. It's already make a publication, and we also make patent from that, and hopefully we can have collaboration with industry, and before probably have to testing it to make sure that it's very good for baby. And we also have studies on tempe oil from tempe with various fermentation times extracted with hexane without without and with pre-washing with acetone and or ethanol, and it gives the result is quite different. So that means that if we want to make this tempeh oil to use for the functional food, we probably can which one that's very good for have a high antioxidant, then can be added to uh, functional food ingredients. And we also conducted Miss Yati Mariati conducted fermented beverage from using kombucha with broccoli and then spinach. And we after the fermentation, we also use the membrane to separate it from the feed, and then the retentat and ferment. And we found out that the antioxidant activity actually in the broccoli show good in permeate. That means it's a small molecule that come out from the from the membrane. That's whereas the other is a retentant, is more the different activity. And we are analyzing this using LCMS, and probably hopefully we can also use the LCMS later to find out what actually when we use the membrane that come out from, and because they also have different activity. And we also involved in the herbal tea studies using the cakar ayam or selaginella, and we try several uh, mistletoe species. That's because it's continuing my research when we are in trying to isolate what the mistletoe is usually used for traditional medicine in for cancer therapy, alternative medicine maybe you call it. And we found we test that from for antioxidant and anti-cancer, and we found when we mix the cakar ayam and risotto and make tea, herbal tea preparation, the activity is better in the mixture. So it is a good, and it's because it's already like people used to, but drink this one. So, but for commercialization, maybe we need to do like the proper uh, toxicity and activity testing. Although for the just for to make a tea, there is already some uh, uh, available in the market. So maybe we, if some will interest in to develop this one, it will be good as well. And because of the COVID-19, there is an interest of this functional food, because like from this study, they show that viral docking entry is reduced by flavonoid or vitamin D3 or either iodide and organic sulfur. And also viral replication is reduced by polyphenol and vitamin C, as well as zinc and its ionophores. And innate immune defense also increased by thymus extract, probiotics, flavonoid, and sulfated polysaccharides. About sulfated polysaccharide, I haven't make the presentation, but uh, this year we are continuing from last year project, the Incinas. Dr. Sofa also make the uh, biscuit for cookies from algae, the marine algae caulerpa, and now we're going to mix in with the microalgae is from to because we found it's have a immunomodulator activity, and one of the is a sulfated polysaccharide that they found isolate from the caulerpa. So it's also like we found that the oxidative stress can be reduced by vitamin C and vitamin D. It can be from the fruit and also fermented fruit, and also the environmental reaction of some botanical and probiotics. And hemostatic immune system also increased by vitamin C, flavonoid, and polyphenol. So for the because this COVID-19, so
Professor,
yeah,
Professor, you have
yeah,
still 5 minutes left. Okay.
yeah. Maybe I finished before that is okay. We got the Miss Yati Mariati involved in making the guava fruit fermented with kombucha that help to increase our immune because it will be produce some the fermentation could be produce some acid that it's not formed if we just make the guava juice. And then for Miss Hani Mulyani and Setiani also try to make the herbs what call it the marmalade that contain, but it's not just the herbs. They also fermented it with lactic acid bacteria, and then make it to the make it a marmalade that can be used for the bread spread or to drink. So it can be both to do both, and this hopefully it also increase the immune system for people so we can avoid the COVID-19. I think I finished my presentation, and hopefully that the functional food that we develop from our Indonesian biodiversity that also hopefully will keep our immune high so we don't get COVID can be together with the there is industry or the we can collaborate with the uh, small medium price enterprises that but this also with good
Parasitical and also natural products. Thanks again for your nice talk. I love herbal tea. I hope you could market the tea very soon. And for the next speakers, so we have Professor Dr. Nurul Huda. Now he is here or not? Yes.
Yeah, yes,
Could
I'm
you
here.
hear me?
Yeah, yes, Dr. Andri, I can hear you.
okay I stopped it.
Hello?
I will introduce you.
Okay.
Yeah. Okay. So due to the poor connection, he cannot present her slide. So you can still listen his talk, sorry, from this Zoom meeting. So we have a last speaker from Malaysia, and he is an expert in food science and nutrition. He is an associate professor, Dr. Nurul Huda. He will deliver his talk with entitled Exploring Application of Fish Collagen as Wound Healing Material. Before he starts his talk, I would like to introduce his background. Currently, Dr. Nurul Huda is an associate professor in food science and nutrition from the University of Malaysia, Sabah in Malaysia. He started this position in 2018 up to now. And from 2016 up to 2018, he was also associate professor in the School of Food Industry, Faculty of Bioresources and Food Industry, University Sultan Zainal Abidin in Terengganu, Malaysia. His associate professor career also began in 2005 in the Food Technology Program of School of Industrial Technology, University of Science Malaysia, Penang, Malaysia. Professor Huda achieved his doctoral degree from the Food Science Program at the university in 2013 and also Excellent Service Award by University of Sultan Zainal Abidin. And recently in 2019, he got Research Exhibition Award from University of Sabah, Malaysia. And ladies and gentlemen, please welcome Professor Dr. Nurul Huda. The time is yours.
Okay, thank you, Dr. Adi, for nice introduction about me. First, I want to sorry to all of the organizing committee and also to the keynote speaker, Dr. Nimal, and also Professor Anita. I expect your presentation must be a very important thing, but I'm not able to join with you to follow your presentation. So actually, the title of my presentation is can I share my slide by myself, Dr. Aldi?
Yes, you can if you could share your slide. I didn't see up to now.
Yeah, I think better if I share my slide by myself, so I can freely by myself. So can I share by myself my slide? Can I try now? Yeah.
Yeah, please try.
Okay. Okay, can you see my slide now?
Still ongoing. I will wait for that. Yes, now I can see.
Yeah, we can see now.
Yeah, you can start from now.
Okay, you can see my slide now. So the title of my presentation is Exploring Application of Collagen as a Wound Healing Material. Maybe some information already introduced by Dr. Niles Normal because we are come from a similar field. It's more fish processing technology. I think I am the close friend of your Professor Benjakul. And also maybe Professor Nina already mentioned something about the cucumber as a potential wound healing material. So maybe some information already with Prof. Nina. So my presentation is focused on the fish collagen because maybe Dr. Nima is quite, I expect is talking with quite rank of the fish protein hydrolysis. So first, let me introduce my place, my university. My university is located in the north part of the Borneo. Yeah, the north part of the Borneo, University Malaysia Sabah. So our university consists of 3 campuses. The main campus is belong in Kota Kinabalu. The International Finance Faculty is located in the one island, Labuan Island, and the Sustainable Agriculture Faculty is located in the area of my current campus. So actually our university is very beautiful university. So I invite once the COVID is we can hold this COVID, and then it's welcome to all of you. I will to come to university to visit my university, and you can talk about the collaboration between my university and your university. So actually my university, my current university is belong to one of the green campus in Malaysia and also in Asia. So the area of the campus is put in the hill, and then we had a beach. So it's a very very good place for the student, for the scholar to develop the knowledge to conduct the research in the respective field. This is my faculty. So faculty I'm come from Faculty of Food Science and Nutrition. So we said from Faculty of Food Science and Nutrition. So in our university we also offer degree in business, computer engineering, humanities and design, psychology, science and natural resources, and also sustainable agriculture and finance. Just some introduction about my current university. So you can go my detail, and then I will come to any collaboration from the participant to explore a lot of things that we can explore together. Please feel free to email me or to WhatsApp me for any potential collaboration between UMS and your institution. So it's an outline of my presentation. First, I want to give some introduction first, followed by phases of wound healing, because in my presentation, I will focus on the wound healing material. The wound healing material, and then followed by some explanation on the existing development of the wound healing science. And then we will enter the most important part is about the potential of the fish collagen hydrolysate as a wound healing material. And then also some sample how to extract this collagen hydrolysate from the fish material, and current study on the wound healing material from this collagen hydrolysate. And we'll end with the challenges and outlook on the potential wound healing agent from fish resources. So this is a world capture fishes and aquaculture production from this figure, and then we can see the number of fish is increased yearly, whether from the capture production or from the aquaculture production. Probably maybe from starting from 1990 is more on aquaculture production, because some problem with the capture production. There are some of the marine fish population are affected, and then we can exploit the marine fish as usual. So why from the 1990 more aquaculture activity is encouraged by government to produce to supply enough amount of fish protein to the people, whether from the freshwater fish or aqua inland water, or from the marine matter or marine fish. So most of the so far the inland aquaculture production is much contribute from the freshwater fish, but I believe for the next decade maybe contribution from the marine aquaculture is more significant. But not all of the fish that we capture or produce from the aquaculture is uses. Some of the fish, especially from the capture, is not suitable for the human consumption due so to some reason. So why some of the food, some of the fish produced most of the fish produced dedicated for consumption, but some of the fish is normally used for non-food uses. And also we can see from this graph the consumption is of the fish product is increased yearly, as in line with the increase of the population. It also some information that every year is around 46 million ton is the discharge in globally due to some problem, due to some, but mostly the problem related with the weakness of science and technology to use this kind of this raw material. So normally this kind of raw material is belong to a byproduct. So normally because normally during the processing of the fish material, probably Dr. Nirmal already mentioned about the byproduct is around 35%. Can be more, can be 50% from the whole body of the fish is belong to byproduct. We cannot use 100%. So far, we cannot use 100%, especially sample from the fillet industry. So filler industry maybe can use only around 40 to 50% from the whole body of the fish, and then the rest is consists around 40 to 50% is belong to byproduct. That we need to think how to use this valuable material to produce more valuable product, rather than we discard to the environment and make some negative effect to the environment, such as pollution. So far the current technology used by byproduct, as mentioned previously, is around 40 million tons. Is available the byproduct, whether from byproduct from the fish processing industry or from the some suitable fish species due to some reason. So of course there are some technology available to use this kind of raw material. Normally use produce some food ingredient. Maybe we can produce from byproduct or from the unwanted fish species. We can produce some fish protein hydrolysis and use ingredient in our food system or the product. But we can also think to use this kind of the product to produce more valuable product with some bioactive compound. We can with some biological active compound, such as with some antioxidant activity, with anti-proliferative activity, with anti-ammonia, antipertensive, and antibacterial. So we have a choice whether we can use this kind of byproduct or the low commercial fish species to produce a feed material or only to produce a food ingredient, or maybe we can choose to use this kind of food material as a raw material to produce the product with high value-added value and come with some biological activity. So in this presentation, I focus on the wood healing potential of the fish collagen as one of the byproduct to produce the product with wood healing property. So maybe some of us we still remember is around 3 years ago. 3 years ago, is quite viral in the internet how the Brazilian doctor tried to treat the bone victim from the torrefied explosion with the fish skin. There is I think it's 3-4 years ago. It's quite viral in the internet about this news, and not only for the human but also applied to the animal. How they use directly the fish skin to treat the burn victim. But then with this idea come to the surface, how to produce, but how to study is more detailed about the potential of this fish skin. Actually, the fish skin is considered as a fish collagen because the main component of the fish skin is belong to collagen, and then to treat the burn victim from exploit or from any accident with the proper way, because it look like not nice if our body is covered with the fish skin. Then we need to think the proper technology to deliver this potential of fish skin or fish collagen as raw material for the wound healing process. This is the idea why currently me and my student working on the wound healing activity, wound healing potential from the byproduct of the tropical fish. So let me see about the phase of the wound healing, because once people experience with the wound problem is that need to conduct the 3 phases for the wound healing process. What is hemostasis? It can be hours, maybe 3 to 4 hours, followed by inflammation, and then proliferation can be a days or a week, and after that restoration. Restoration can be a month. This is a process from the wound healing restoration. Then each phase of wound healing process is influenced by a series of essential mediators. So it's quite a complex process. So let the medical doctor focus on this process. So we focus on the accelerator material. So because in this process is influenced by a series of essential mediators, platelet, synchronized inflammatory cells, cellular, extracellular matrix, protein, growth factor, inhibitor, and accelerator material. And collagen is part of the accelerator material. So we focus on the accelerator material. So besides the collagen as accelerator material, why is the collagen come with a potential for the accelerator material? Because the collagen fiber is a major component of the skin. Is a generally known. And then if we directly use the collagen, essential process is much faster because the collagen is there. Our skin is need a collagen for the wound healing process. And then we expect the wound healing process will be much faster if we can provide sufficient amount of the collagen from the for the wound healing process. So beside from the collagen, previously some scientists also report the accelerator mediator not only exists from the collagen, but also from the fish oil. So fish oil also can play a role as accelerator mediator for the wound healing process, as mentioned by Karom in 2000. So we from fish oil we can also use the ointment for mecococcus as accelerator material, and also from the curcumin for the conjuncted with hyaluronic acid. So one of the potential accelerator healing material, and also catfish and the aloe vera, honey, and milk ointment for the secondary bone impact. And then you can see from this picture, this one is without any. This one is control group. One is belong to control group. Control group. The animal is treated by saline solution only, and then the group 2 is treated with with ulcer, simple design is a commonly treatment of the bone people, and then the group 3 is belong to the honey, milk, and aloe vera ointment 5%, and then we can see that treatment with honey, milk, and aloe vera is able to close the wound is much faster, is much good, better than the control one, and also compare with the origin and silver a little bit at 3%. So this one also another evidence from the curcumin, from curcumin conjuncted with hyaluronic acid. So hyaluronic acid we can extract from the heat of the chicken, from the red part of the heat of the chicken we can extract the hyaluronic acid. So you can see this one from the graph by Dr. Sharma. We can see the untreated one. The untreated one is a one close to is quite low. It's only around 25%. But if we treat with the animal with curcumin, and then the wound closure is much significant higher increase. And so if we if we treat with hyaluronic acid, if we combine together curcumin and hyaluronic acid, and then the wound closure is almost 100%. So the next evidence also from the fish oil. So meaning we can use a different kind of the product to as a wound healing material, wound healing agent. This one from the oil from the fish head, and then you can see from this result. So if the one comparison flora, so you can see from this graph. So within the 11 days, so the totally 100% of the wound is totally closed. If we treat, if the animal treat with the fish head oil, comparison control one, and also comparison commercial flora. So and then let me see the collagen, and let me see the collagen because I mentioned because they mentioned previously that actually the skin is consists collagen. So the similar structure with our skin. Our skin also consists collagen. So actually, according to collagen is actually from the root word is a glue and to produce. So actually, the collagen basically explore because it's a component tissue when we boil and to produce glue. And after that, some development of the collagen science and technology. And so far, the 29 distinct collagen type have been characterized. So collagen is considered a different type. So far, the 29 collagen type is characterized. Then all of the collagens, 29 collagen display are typical triple helix structure. So what is the trademark of the collagen is this collagen is consists of the proline and hydroxyproline. So normally the structure of the alpha chain of the collagen is consists a sequence of the glycine, proline and hydroxyproline. So why normally in order to determine whether the material come with high amount of the collagen, we can conduct analysis based on the content of the proline and hydroxyproline, what we call as amino acid. So normally raw material come with high proline and hydroxyproline, it also come with good property of the collagen from the gel. This one also, however, they mentioned to you 29 type of the collagen is characterized, but the only 9 from the 29 is only most belong to the most common collagen type. Is the 9 most collagen type is available in this raw material. This one just inform to participant about the collagen. Meaning, if you talk about collagen, it's not only collagen. There are 29 type of collagen is available, and then we need to understand the structure, the characteristic of each collagen in detail to know more knowledge about collagen. What will happen if we experiment with the animal or people experience with the wound? What we say wood accident. So actually, if the healthy collagen is come with the triple matrix, and this one is a healthy collagen. But if we experience with wound, some damage is take place. And sometimes it takes place, and sometimes it takes place in the triple helix. And then with addition of the collagen from outside, small molecule of the collagen, it will be able to faster, quick recovery of the damaged collagen. This is one of the scenario is proposed by scientists, how the gen peptide is able to accelerate the wound healing process due to the small peptide of the collagen from the outside. As I mentioned previously, we have a lot of fish, especially in Indonesia, with 70% is consistent marine. A lot of fish species available there, and then we need to explore together the potential of the fish species as a wound healing material. So we can start this collagen from the marine fish, especially from the byproduct. Byproduct, I mean, consider from the skin, from the bone, or from the scale, and to produce a collagen. So when we produce a collagen, and then we need to hydrolyze a collagen. We need to go to next step to produce a collagen hydrolyzed. And then when we produce a collagen hydrolyzed, maybe with a different property, which depend to the process, depend to the enzyme applied, depend to the acid we use during the extraction. And then we can apply, we can test the produced collagen hydrolyzed as a wound healing agent, whether in different form, whether in the sponge form, cream form, hydrogel, or spray. So far there are less knowledge, less report on the potency of the fish collagen. There are some report on jellyfish, Alaska polar, tilapia, salmon, haddock. But all of the reports show the acceleration of the wound healing rate by using this kind of the collagen hydrolyzed. But you can see from the list of fish, most of the fish is not belong to tropical fish. Most of the fish is belong to temperate fish. It means consider maybe if we need to explore the potential of our tropical fish as a raw material to produce the collagen hydrolyzed with wound healing used by the fish. It belong to the properties. So it's a way, it's a one, it's a extraction method that we can apply. It's a one of the methods that we can apply to extract the potent fish collagen hydrolysate. So by fish spray product, whether from skin, bone, or scale, and we need to conduct for pre-treatment to produce the acid soluble collagen. We need to use remove the non-collagenous material. So we need to reduce the S content by using the DT. We also need to reduce the fat content by using maybe using the butyl alcohol if the fish skin come with high fat content. And then we produce the acid soluble collagen. So acid soluble collagen, we call it acid soluble collagen, and then we conduct the salting out, and then we produce the acid soluble collagen dry form of the acid collagen. But during the production of acid collagen, there are some pellets, precipitate. So the precipitate we can extract again. We can extract the precipitate by using the enzyme to produce the peptide soluble collagen. And then we freeze dry. And then they produce the soluble collagen, whether from the acid or peptide, and then we need to hydrolysis to produce a small peptide. To produce, whether we can apply any enzyme, whether collagenase, papain, alkaline, potassium. So we can use many type of enzyme. And then we can see which enzyme suitable for our raw material. And then we able to produce peptides or collagen hydrolysate, and then test to we connect to conduct the characterization and test to the animal. So this is a current study by scientists. They're using, as mentioned to you, most of the wound healing by controlling the inflammatory reaction and increase the wound antigenesis and collagen deposition. So similar report also on jellyfish. Jellyfish improve the structure closure is much faster than the control one. As Alaska polar skin, Alaska polar can enhance the wound healing activities by oral administration in animal study using the rat. And also from the unicorn jack skin and also to Nile tilapia skin. All of the report come to with the conclusion the potential of the fish collagen as a wound healing material. But unfortunately, as mentioned previously, all of the raw material fish species used is not belong to the tropical fish. It's all belong to the temperate fish. And then it's our time now. It's our time now to explore the potential of the byproduct from the tropical marine fish or tropical freshwater fish as a wound healing agent. So also from the giant cockle, similar cuttlefish, blue shark, and also haddock, and also chum salmon. So there are the report that mentions that support the theory that the fish collagen is able to act as a wound healing material. This is a detail report from the Dr. Ying Chen about the ability of the giant cockle collagen for the wound healing. Yes, you can from this ability of the animal with wound experience. So you can see from this part, the wound closure is much faster if we treat with the peptide collagen, percent soluble collagen rather than the control one. Control meaning they use a saline solution to treat the animal. To similar report from the Morad Mandiri by using the collagen from the cuttlefish. Jadi from the cuttlefish skin, and then extract the collagen hydrolysate peptide, and then treat to the animal. So you can see this one is the group 1 is belong to consider the control one, group 2 and group 3. So as you can see how the cuttlefish collagen hydrolysate able to close the wound is more quickly rather compared to control group. A similar report from the shark fin. from the shark skin. Shark skin also consists of collagen as to promote the impression of the wound healing. We can see similar report as reported there. A similar result so the potential of the collagen hydrolyzed to treat the animal with a wound experience. This one also the similar report using the salmonella and also tilapia negotica skin, how this collagen extract from the salmonella skin and also from tilapia skin is able to improve the healing rate of the animal if we treat with this kind of the collagen. Similar report from the top pellet. So a lot of evidence, we have evidence that the ability of the collagen from marine fish as a agent from the wound healing process as accelerator during the wound healing process. So this one also from the Alaska polar. Scapola is a raw material to produce a surimi. The main raw material to use a surimi production is belong to Alaska polar, and then people can use the byproduct from the Alaska polar skin as to produce the collagen hydrolyzed and come with evidence that the collagen hydrolyzed they produce from this Alaska polar skin is able to come with an alternative for the wound healing treatment. Similar thing from the salmon, come with the evidence that the collagen from the cell scan of the salmon also can act as a good potential wound healing material. So this is a challenge now for us in Indonesia with a very huge amount of the fish species to explore our own fish as a potential raw material to produce a collagen peptide, collagen hydrolyzed, and then to see whether the collagen peptide produced for our tropical fish able to come with a good property as a wound healing agent. So a lot of fish species now we can use such as a lizard fish, big eye snapper, or Nemipterus japonicus, scrisi, or maybe different. It belong to godfish. It's a godfish. All of the fish is belong to underutilized fish. Then during the surimi production, they also produce about 40 to 30% of the byproduct. So we can use the byproduct which consists skin, scale, and bone of this kind of fish as a raw material to produce a collagen hydrolyzed or collagen peptides, and then we can test as a wound healing agent. So this is my current PhD student. Yeah, we just started this project last year, but due to the pandemic, our result is not come yet. So we're not able to produce any publication yet related about the potency of the tropical marine fish. So we're working with a different fish species is available in our tropical country, and then study the abilities, the characteristic of the peptide hydrolyzed as a wound healing agent. But the study is on the way now. It's quite slow due to the pandemic, so we've not entered the lab really. So hopefully after just after next year we can come with the evidence. So we still keep the raw materials that we use, but we try to use some potential tropical marine fish for our study. And then it's all my presentation about the potential of the fish collagen hydrolysis as a wound healing material. Okay, thank you to Dr. Adri.
Thank you very much, Professor Nurul Huda, for your nice talk. Very interesting topic related to collagen from fish sources. And now we are for the next session, which is question and answer. There are several questions in the chat room, and yeah, so all the speakers are ready today, and I will see the question in the chat room from the first one. Oh yeah. The question is for Dr. Niles Nirmal from Dodi Dharmajana. Are there any side effect of FPH as a medicinal ingredient? Maybe you can start to answer this question first. Are you hear me, Dr. Niles Nirmal?
Yeah. So, well, what is the question?
Again, the question is, are there any side effect of FPH as a medicinal ingredient? From Dodi Dharmajana.
Is there any what?
Side effects.
Side effect. Yeah, side effects of
FPH. Fish protein hydroxylate. Yeah.
See, like as I told, this is from the natural source, like whatever we are eating, like sometimes fish protein hydroxylate from fish place, and sometimes from the waste product. So it has side effect, but not really harsh, and it's again depend like whether you are applying it like from outside is not, but if we take as a supplement, it's depend on the human body. It's depend on each participant or each person, how they respond to the external protein source and everything. Am I answer the question? Hello?
Committee, can you show the Dodi Dharmajana? Maybe is it okay for the answer?
Hello,
or maybe I think yes. If he has more question, he will put in the chat.
No, as a medicinal ingredient.
Yeah. So we go to the next question question for from Sri Priyatni. According to your information about FPH, do you have the project for large scale of FPH and what application that you have done?
Yeah, I think I have replied everyone through chat box, but I can reply here again.
Yeah, because maybe everyone want to know also for the answer of this question.
So yeah,
maybe they have the same question. Yes.
Yeah, we are now in the process for applying a big project, and yes, our past work shows that ACE and DPP inhibitory activity in our laboratory study. Yeah, these are the like applications in the sense of various biological activities, but we haven't did any clinical trials or anything yet.
Okay. I think the most of question is for Dr. Niles Kumar, but I also want to ask from maybe Professor Artanti. Are you hear me? Hello, Professor Artanti.
I think she's not there. She turned off.
Yeah, maybe I will give you one question from me. What is the main challenge as a researcher in Indonesia to mine the potential of Indonesian biodiversity? The main challenge of a researcher as an Indonesian researcher.
I think the main challenge is that we have to, if we can, if we want to go to the industry, usually it's better if the industry is.
Can you increase your voice? Sorry, I couldn't hear.
As a researcher, I think not just in Indonesia, if we done research and we expect the industry to come to us, it's a bit difficult. But if we already have a talk or something, and then what they want, and we do the research for them, it will be useful, because sometimes the industry know better which like functional food that will be booming or something compared to us as a researcher. We just we try this, we try that, but we don't really know what like expectation from the people, because that's why we are not become we don't have an industry. That's why I think that's the challenge, is to know what the industry want research one. So I think we better collaborate since the beginning, what they want,
Yes.
and we do the research. Especially now, if we get already the facility, and they because sometimes if we do research, but then we don't have the industry who will take our research, then it's just that it's just probably just become a publication. Although we do make patent, but probably no one will buy our patent like that. I think because it's probably we as a researcher, especially for working with science, we are not really know what like people actually like now in COVID. We know that everybody now making like the functional food that increase immune system. But which one that will be booming? I think we don't have that idea for that. But the industry maybe have that.
Okay. Maybe in the future we can do collaboration with Thailand, also Malaysia here. They
Yeah,
Yes.
will
yeah.
make a bridge for
So
us to make a
I would like
collaboration.
to, yeah, I would like to give
Yeah.
my opinion and my experience in this area as well.
Yes.
So when I was in Australia, I worked for the Australian native plants, herbs and fruits. So indeed, these native plants are really, really good. Of vitamin C and some other phenolic compounds. So I work on a Davidson plum and other, which has really high antioxidant activity and very high anthocyanin content. So I work for like herbal tea for two of the product from Australia, indigenous Australian plants, which is really exciting and fascinating. And yes, these indigenous people, they are drinking this every time, and it really give you boost. I believe like each country have um, their indigenous plants and everything. That knowledge should come to the public. So we really have to go and meet our indigenous people, or these people like native people. They have that knowledge. We have to get that knowledge from them, and we have to translate through research into the public awareness to make them interest. Yeah, this is really good topic.
Okay.
Thank you,
Thank you
Dr.
so much for your.
Dina. Yeah.
Okay, thank you.
Yeah. Okay. So we start. Also have another question in the chat room. Oh, maybe this question is for Professor Dr. Nurul from Any Soliha. Dr. Nurul, you hear me?
Yes, Dr. Adi. Yes, I'm here.
Okay. Yeah, the question is there any effect of freezing treatment on enzyme hydrolysis process?
Okay, thank you, Dr. Andri. Thank you for the good question. I think as mentioned, we are just on the way. I just start this project last year. So we received the highest research project approved by my university. Is with value is 600 juta rupiah. Yeah, the total budget for this research is 600 juta rupiah, but start last year only. So and then during the pandemic, we're not able to come with more result now. We know more result now because the one pandemic COVID coming, the university closed and they close again. But we expect for the for the if the piece is frozen piece or fresh part of to the property of the collagen hydrolysis. Okay, I think it's
Okay.
my answer, Dr. Adi.
Yes. Okay. So there are still numerous questions. I will read one by one. Where is it? Ahmad. Sorry. Okay. There also another question from Ahmad Fatoni for Dr. Niles.
Yeah.
Thank you very much for sharing a lot of information regarding FPH and its biological activity. According to your experience, what are the challenges in research?
Yeah. So the main problem is consistency and reproducibility. That is the main challenge in this production of FPH, fish protein hydrolysis. Yeah, even the fish source, even if we consider one species, the one species does not mean they have the same proportion of protein content or their body composition. So that make quite difficult to produce, get the consistency in the production, and get the like same reproducibility or uh, batch to batch quality of the fish protein hydrolyzer.
Okay,
Yeah.
thank you. We still have more questions. I hope
Thank
you're
you.
still okay with this question.
Yeah,
So
yeah.
another question is from Wita Kartika Restu
Yeah.
to Dr. Niles. I'm interested in short peptide from FSH and would like to ask regarding the how much the yield that you get from the raw material of fish until the purification to be the targeted short peptide. Have you ever used the short peptide for other functions, such as the hydrogel or other any form,
Yeah.
as some of the hydrogel are also aimed to kill the cancer cell line?
Yeah. Thank you for the question. See, yes, actually, indeed, if you go for to purify any compound, okay, the yield is very low. It could help uh, like anti-proliferative or to kill the cancer cell.
Okay, still have more question for you because most
Yeah,
of
yeah.
people interested with your
Sure.
talk. And the next is Koirun Nisa from LIPI. What kind of parameters and instrument do you use to confirm that your FPH product is successfully produced?
Yes, so usually how to confirm that? So the first thing, like if you check the biological activity of your raw material and the fraction, you can see there is something like obviously the raw material or the first fraction may not give you any result, but the fraction can give you the result. So it shows that something has happened. Something has happened to the raw material, which is the long polypeptide chain. It has degraded, and you get the some fraction. Something has happened. So next step, what you can confirm is again you can go for the chromatography and something that and get the fraction, and they check the biological activity of each fraction. Furthermore, you can go for the mass spectrophotometry, which can give you the molecular weight. From there, yeah, you can yes, I get the hydrolyze it or the smaller peptide compared to the
Okay, thank
If
you.
the material come from the area from the nearby to the mining area with some what great expectation that some heavy metal content is quite significant, that yes, you need to conduct the heavy metal analysis first. So because we need to make sure that raw material is used for the production of the fish collagen or collagen hydrolysis is belong to safe raw material. Otherwise, maybe the raw material is not suitable, is not qualified to use any process, not only to produce the fish collagen hydrolysate or the hydrolysate for normal consumption. It's very important if the area are decontaminated or nearby the mining area. It's very important to conduct the primary analysis of the heavy metal contamination, and especially according to some previous research, if the fish around the area with contamination, the heavy metal content in the skin. So the accumulation will take place in the skin. So then if we use the skin to produce a fish protein fish collagen hydrolysis, of course, the heavy metal will start come here. So we need, it's very important to conduct the preliminary analysis that the raw material that we use is considered safe for human consumption. I think it's my answer, Dr. Agli.
Okay. And we still have a question for you from Annie Soliha. Is there any effect of freezing treatment on enzymatic hydrolysis?
I think we have
Oh, I think yes. Sorry, we have to typing. So, okay, we start moving to Professor Artanti.
Okay.
There are different using. Are there any different using food between for functional food and for drugs, such as for lowering cholesterol?
I think when you say it's like different from food and drugs, usually for the functional food, used to generally eat food. So if you use it as a drug, usually you extract them, you boil them like for until from 1 liter become only small amount. So I think the difference is in the dosage. Usually, if you want to use it as a drug, usually you use more compared to when you if it's already have lowering effect, even though you just drink like the herbal tea. If you try to do the DPPH directly from the herbal tea, it seems that you have acidity a bit high, but you drink a lot. You drink like at least 100 ml of tea compared to if you drink drug that only 5 milligram or something. Maybe the 100 ml of tea is the same as the 5 milligram drug like that. But then that's why it's the different how to you take it. I think it will be same, but it in different way. Is it answer your question or not? Because usually when you
Yeah,
work
if Mhm.
with food, it will be like you measure the IC50. It won't be very low because if it's too low, then if you eat too much, it will be dangerous like that. So if it's for antioxidant or anti-cholesterol, usually that's it's because you also it's like you eat the whole fruit that's like maybe for lowering cholesterol, what kind that you like that. But we know
Mhm.
and we do the extract, so it has the activity for the lowering cholesterol. But usually, if we because it's eat as a food, so the concentration won't be.
Thank you so much for Professor. And we have still have more question from Annie Solikha to Dr. Nilesh Kumar. Based on your research, which is the better method for producing FPH?
All right. So better method is enzymatic hydrolysis. The enzymatic hydrolysis can give you really um, small molecular peptides. So that is really good.
Mhm.
However, again, it's depend on the what is your interest and something. And I also explained like fermentation hydrolysis, which can give you two benefits. Like it can produce protein hydrolyzate and it commercial applications. So these are the yeah. So obviously depend on your aim. If you really want to go for small molecular weight peptide, enzymatic hydrolysis is the best production process. Yeah.
Okay, thank you. Still have question for you from Ahmad Sarifuddin Lipi. The
Yeah.
question is, okay, nice presentation, Dr. Nirmal. Thanks for sharing your knowledge. I ever heard that FP8 has functionality to prevent high blood pressure. That
Yeah.
is very great news for high blood pressure patient.
Yeah.
But in terms of application, because most of Indonesian or common people don't like fishy taste and
Yeah.
aroma of FPH, is
Yeah.
there any way to get rid of this off flavor of FPH? Yeah.
Yes. Yes. We have a good news. There is a way that we can avoid the fishy smells, and it can make like a plain without any smells, or we can add some other flavor which can improve the aroma and everything. There is a process called debittering process. We can use that as well. Yeah. So
Thank you. Is there
yeah.
more question in the
Yes.
chat or not? I will check again. Maybe I have a question for Professor Huda. Professor Huda there, are you there?
Yes, please.
Yeah, I just want to know, is there any difference between the product, a collagen product from marine fish and freshwater about the quality or something else? Thank you.
Okay, thank you, Dr. Adi. I think each fish come with its own property. Each fish come with
Mhm.
unique property. So we need to study species by species. We cannot generalize all the fish species is as one category. So why maybe some species is potential, maybe another species is not potential. So in general, each species come with its own property. So we need to study species by species, and also not only because due to the different species, but if we conduct the process differently, so the result will be different. If we use a different enzyme, and then the result also different. So why we need to specifically. So as we need to work specifically to the species and also to the specific enzyme to produce a better quality of the collagen hydrolysis. I think it's my answer, Dr. Ali.
Thank you so much for your answer. How about from Dr. Nile's perspective about this also? The question is from collagen, right? Collagen quality from different source like marine source and also the fresh water. How about the hydroxylate, fish protein hydroxylate? Also the same quality, or maybe you have another perspective to answer?
Yeah,
Yes.
as Professor Nurul already told you that yes, every species has different compositions, so we have to study that. And obviously, as I also answered the previous questions, like the raw material is affect your final product quality. So we have to do that. So this is the basic challenge in this, although like Professor Nurul already introduced you that collagen has a different types, but if you can able to successfully isolate or focus on one particular type, then that is the different case, but yeah, the composition of sea and fresh water is different. Yeah, thank you.
Okay, thank you so much. Is there any more question in the chat or not? Maybe if still not, I have a question also for, oh yeah, we have one more question. Okay, it's different things. Maybe I have a question for Professor Artanti. I'm so interested with the tuber diversity. Where can find the tuber diversity in Indonesia? Is only from the west of Indonesia or from all the part of Indonesia? Usually, yeah.
All part of Indonesia maybe have the different kind of tubers, but the problem is we don't really use it. So we like now people who eat sweet potato now also grown that when the wild type probably still there. So I think probably the biology people who's doing exploration know the centra of the, but because people don't grow it that much, then it will be difficult to get it because only very limited people who have this, and probably we can lose the genetics if we don't use it like that. Because I think we most usually use the already domesticated species for that's why then probably the like the black rice, they probably have other things like I think some from biotech doing the exploration from to get that sometimes what protein or something from the black rice. Now we are aware of that, but then sometimes we aware when we like this COVID situation. But then when this situation is normal, then we just something. But if you already do organic, is it really enough to support the because it will be down, I think, the production. That's why it's still using something like that. So that's why we I think since I was first in LIPI, oh, we have to do diversification or something, but it seems like still only for the small part of research, but not really people use it, something like that.
Okay, thank you so much for Professor Artanti. And I think I did not see more questions in the chat. So I think that's all for the second plenary session for today. Thanks for all your contribution to the discussion. Also, thanks to Dr. Nirmal, Professor Huda, and also Professor Artanti for your valuable time at this conference. See you other times. And now I will give back the time to Dr. Marisa. Dr. Marisa, the time is yours. Thank you.
Yeah,
Yes.
thank you.
Yes, Dr. Andri, thank you for the nice leading in the sessions. And also we are thank you to all of the plenary speakers, Dr. Niles Nirmal, Dr. Professor Nurul Huda, and also Professor Nina Artanti, PhD. Well, ladies and gentlemen, we have several agenda for today, but because now we have a complete speakers and also I think we are still have a complete participants, it's better for us to get screen captures. So please, all of the participants and our honorable speakers, please turn on your video. The committee will take this will capture the screens. Okay, we're waiting for all of you to open the video. Okay, so thanks to all of participants that I think when I see the list of participants, is in this day, the same with the first day of our symposium. We are really grateful that you still attend our symposium till the second day of symposium. I will tell to you, all of you, about our next agenda. The next agenda, we have two parallel sessions. Will be start from 1 p.m. until 3 p.m. The first parallel session will be divided into two sessions, session 3 and session 4. The session 3, the first room of parallel session will be discussed about natural product and traditional medicines with the moderator Andi Febrisiantosa, PhD. And also the second room will be discussed about analytical chemistry
Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave uses high-temperature steam as a sterilizing agent. Then, in the filling step, gudeg are filled in cans. The next step then continued by exhausting and steaming process. After, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, consisting of people and population, culture and language, policy and governance, or good governance, local government and regional autonomy, social conflict, economics and poverty research, scientific services, consisting of scientific repository, metrology and standardization, innovation of technology transfer and utilization of science and technology services. studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world, and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Ecohydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions.
Bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Each vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817 with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bam.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Gagus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda dan mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian. Sekian dari saya. Salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta. the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada semuanya di acara International Conference
Festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISE 2019, Econ Probios, one of the member ISE conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISE 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISE 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudek in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by Infinity Science Exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Ilmiah Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri dapat menjalin kolaborasi berkelanjutan untuk pemanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of good manufacturing process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Dunia Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Secara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat,
particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefit from interacting with various scientific stakeholders in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and brongkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, brongkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Dunia Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu 1. Saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholders in that event. However, now we got different conditions. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with addition side dishes such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam as a sterilizing agent. Then in the filling step, gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi wabarakatuh. Perkenalkan, nama saya Agus Ketut Sunardianto, peneliti dari Pusat Penelitian Dunia Lembaga Ilmu Pengetahuan Indonesia. Saya sekarang berada di Belanda, sedang mengikuti program postdoktoral saya di Delft, Belanda. Pengalaman saya mengikuti konferensi ISAC 2019, yaitu satu, saya diberikan kesempatan untuk mempresentasikan hasil penelitian saya secara efektif, di mana dari presentasi tersebut saya mendapatkan umpan balik dan komen dari para peserta lain, di mana umpan balik tersebut sangat bermanfaat bagi saya untuk menimbulkan ide-ide baru melanjutkan penelitian saya. Kemudian manfaat yang kedua adalah dengan menghadiri konferensi ISAC 2019, saya juga diberi kesempatan untuk melihat secara langsung bagaimana cara berpresentasi dari peserta lain yang bidangnya berbeda dengan saya atau bidangnya sama dengan saya. Oleh sebab itu, dari situ saya mendapatkan pengetahuan baru terkait dengan topik-topik penelitian terbaru dan terhangat di Indonesia. Kemudian yang ketiga adalah konferensi ISAC Internasional 2019 merupakan ajang bertemunya para peneliti, kemudian para dosen dan juga akademisi serta dari kalangan industri, sehingga tercipta kolaborasi yang sangat kuat yang harapannya bisa membawa dampak kepada masyarakat luas dan juga mengembangkan ilmu pengetahuan itu sendiri. Demikian dari saya mengucapkan selamat datang kepada Bapak Ibu semuanya di acara International Conference ISAC 2020. Semoga presentasi Bapak Ibu semuanya lancar dan acara ISAC juga lancar, sehingga nantinya selain mendapatkan ilmu yang bermanfaat, Bapak Ibu juga dapat menjalin kolaborasi berkelanjutan untuk kebermanfaatan ilmu kepada masyarakat luas. Demikian dari saya, salam ISAC 2020. Wassalamualaikum warahmatullahi wabarakatuh.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in Indonesia. There are many scientific activities in this event, such as conferences, exhibition, and scientific competition, particularly for young people. In ISAC 2019, Econ Probios, one of the member ISAC conference, got many benefits from interacting with various scientific stakeholder in that event. However, now we got different condition. In ISAC 2020, we cannot enjoy directly, but it held in virtually. So, stay healthy in COVID-19 pandemic situation, and you still got benefit from ISAC 2020 by infinity science exploration. Thank you.
Yogyakarta, the city of education and culture, has its own stories and romances in every corner of the city. Each corner will bring us back to the memories that make us to always come back to the city of yearning. Yogyakarta has some traditional foods such as gudeg, mangut lele, lombok ijo, tempe bacem, and bronkos, which are findable in street food stall, lesehan, angkringan, home industry, and restaurant. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package 42 kinds of traditional foods from all over Indonesia, such as mangut lele, lombok ijo, tempe bacem, tempe kari, bronkos. You haven't visited Yogyakarta if you haven't eaten gudeg. The cooking process of raw jackfruits using a stirred process was called ngudeg in Javanese. So, the traditional food produced was then called as gudeg by people of Yogyakarta. Gudeg is served with gudeg are filled in cans. The next step then continued by exhausting and seaming process. After being tightly sealed, the cans go under another sterilization step using autoclave. This sterilization process is an essential step for the canned foods. After the sterilization process, canned food products are cooled down at room temperature and then quarantined for 2 weeks in sterile room for quality controlling. The cans are labeled and ready to be sold. The implementation of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
Assalamualaikum warahmatullahi
Halo. Aku harus mulai tapi kayaknya bentar. Masalahnya aku sendiri nggak dengar diriku ya.
Enggak. Kamu perasaan aja itu. Lanjut aja. Rasanya kita emang nggak dengar. Coba aja.
Halo. Can everybody hear me? Halo.
Hello. Yes.
Bu.
Yes.
Yes, okay.
Yes, we hear you.
Okay, thank you. Because I hardly can hear my own sound, that's why. We are now in the parallel C, food and functional food. My name is Anastasia Fitriadevi. I'm a food scientist from Research Center for Chemistry, Indonesian Institute of Sciences. I'm your moderator of this session. First of all, let me announce the rules. 1. Please have a stable internet connection. 2. Make sure that your computer has a working camera and audio device. 3. Please ensure that the chat room is only available for delivering questions to the ongoing discussion in the session, requesting for changing or moving to the other breakout room in the parallel session, and announcements delivered by the organizing committees. Rule number 4. Don't forget to fulfill the list of attendees that we share in the chat room. 5. If you would like to change the room, please inform our co-host and they will assign you to the room of 2 parts. The first part will consist of 4 presenters and then followed by Q&A. After that, we continue to the second part, which consists of 3 presenters and then also followed by Q&A. Okay. I already checked for the first part. All 4 presenters are already standby, so we can continue. Without wasting more time, let's start the first part. The first presenter is Mr. Dodi Darmajana. He is a researcher from Research Center for Appropriate Technology, Indonesian Institute of Sciences. The title of his presentation is Study of Isothermic Absorption Model of Moisture Content of Fibrous Instant Corn Rice. Let's see the presentation.
For Mr. Dodi,
Co-host,
Astrid.
Sorry,
of
we cannot.
Sciences. I will present the result of our research entitled Study of Isothermic Absorption Model of Moisture Content of Fibrous Instant Corn Rice. The background of this research is corn is a strategic commodity that can be developed into an alternative staple food. It can be seen from the nutritional value of corn. The main nutrition content of corn is starch of 72 to 73%. With 25 to 30% of amylose and 70 to 75% of amylopectin. Simple sugar level of corn are glucose, fructose, globulin, proline, glutenin, and non-protein nitrogen. Instant corn rice is a product that can be developed from corn and a type of food product that is easy to serve. Indonesia's tropical climate with an average relative humidity above 60% increase the possible of air absorption and will affect the shelf life of dry food. High moisture content in food from agriculture product will degrade the quality through a series of physical, chemical, and microbiological processes. This study aims to study the water content equilibrium model of instant corn rice, and the resulting model will be useful for storing instant corn rice in various conditions. So the step can be taken to prepare the storage room condition as well as for the instant corn rice drying operation. For material and method, the main ingredient used in this research are fibrous instant corn rice, distilled water and chemical salt including potassium carbonate, sodium nitrate, sodium chloride, ammonium sulfate, and potassium sulfate. The tools needed in the study are a mercury thermometer, trisensometer, analytical scale, slicer, oven, dryer, Pyrex excavator, temperature incubator, glass clamp, and stopwatch. The absorption experiment was carried out by static method at a temperature of 30 degrees Celsius and 5 times the relative humidity level, 40% to 90%, and that the adjustment was carried out according to the Carey, Hasley, and Alcin equation model. To obtain various constant value in the equilibrium water content equation, experiments were carried out by storing fibrous and instant corn rice in storage container, and the error in the storage package is RH adjusted by adding chemical salt. And in table 1, we can see the result of comparison between the observed RH in the storage container at 30 degrees Celsius saturation chemical salt and the theoretical RH. In table 1, the deviation value runs from 0.4 to 3.8%. And this value can be affected by the purity of the salt solution used or the fluctuation in temperature and humidity that occur around the measurement time. Based on ASTM standard 50 parts, deviation measurement may reach 4 to 5% RH, and according to Labuzet, to get good result and accurate, the purity of the salt solution that is used need to be considered in determining the equilibrium moisture content. For table 2, it can be explained that the higher relative humidity of the surrounding air in package and the higher water content equilibrium on instant corn rice. If instant corn rice is stored in an environment with temperature of 30 degrees Celsius, with RH as high as RH of air at second column, and the instant corn rice water content will run out and show in column 3. And from the experiment result obtained water content of instant corn rice equilibrium in the range of 4 to 16 days. The initial moisture content of instant corn rice is 7.90% dry basis and increased to 8.90% at RH 47.4% and reaches 22.65% at RH 95.7%. The increase in water content will continue until equilibrium with the surrounding air is achieved because according to Hall research, the migration or flow water from a place of high vapor pressure to a place of low pressure is proportional to the difference in partial pressure of water vapor. And the movement of water during the isothermal eruption process can be caused by several factors, include relative humidity, water activity, airflow, velocity, diffusivity, capillary action, and gravity, evaporation, and condensation. In table 2, show the equation model of isothermic absorption and its MRG value from 3 models, Hasley, Carey, and Alcin. If we compare the isothermic absorption curve of fibrous instant corn rice from the tree model, with the experiment model show at the figure 1. And at the figure 1 show that separate model can accurately describe the phenomenon, and the Curry and Alcin equation model can be used to predict the isothermal absorption of fibrous instant corn rice, indicated by MRG value of less than 5, which means the correct absorption isotherm model, and the model chosen is the Alcin because the smallest MRG value of 3.7460, and the Alcin model was chosen to describe the true state of the fibrous instant corn rice isothermic absorption. For a conclusion of this study are the water vapor absorption process occurred in the fibrous instant corn rice isothermic absorption experiment at 30 degrees Celsius, which was marked by an increase in water content with a 40 to 90% relative air humidity treatment, and a balanced moisture content was achieved at interval between 4 and 60 days. And the Austin equation is the best equation model with the highest accuracy because it has the smallest MRG value, and instead the model equation has not appropriate because it has MRG of 5. Okay, well, that's all our presentation. Thank you for attention. Wassalamualaikum warahmatullahi wabarakatuh.
Mechanical properties of edible film from tanduk banana or Musa corniculatarum pills for good packaging. Co-host.
Assalamualaikum warahmatullahi wabarakatuh. Good day, ladies and gentlemen. My name is Murieti, and I am from Polytechnic Negeri Jakarta. Thank you for letting us be part of your event in the International Symposium of Applied Chemistry 2020. We appreciate the opportunity to share and present our research about mechanical properties of edible film from tanduk banana peels for food packaging. This is the outline of my presentation. I will start with introduction, objective, methodology, result, and conclusion. Currently, the extent of plastic use in Indonesia as food packaging material in order to meet daily needs is very high. Plastic is flexible, economical, durable, and good barrier against oxygen and carbon dioxide. But plastic also has several disadvantages derived from petroleum. Plastic degradation is very slow in nature. It would take years for plastic to full degrade and resulting in build-up of plastic waste which causes environmental pollution. Currently, the development of food packaging technology leads to environmentally friendly, biodegradable, more natural, and without preservative packaging material. This also has been conducted to develop biodegradable packaging that is made with natural ingredients that are safe for consumption, doesn't contaminate food product, can maintain the freshness and durability of the product. Edible film has considered as an alternative as food packaging due their capability to extend the shelf life of food by reducing moisturizer and oxidative reaction rate. One of the natural ingredients used in making edible film is pectin, extracted from banana peels. The formation of edible film for pectin has advantage is that the surface smoothness in resulting film, but it has low mechanical properties. Adding glycerol or sorbitol as plasticizer can make edible film more flexible, smooth, increasing permeability toward water, gas, and soluble substances. In this research, the edible film was made from tanduk banana peel pectin with the addition of glycerol, CMC as plasticizer, and tapioca, which expected produce a mixture of edible film as food packaging material with optimum mechanical properties such as tensile strength and elongation percentage. The objective of this research is determine the mechanical properties of edible film. The material and instrument used in this research can be seen here in this slide. A research methodology of this study comprised of 3 stages. The first stage was extraction of pectin from banana and then sieve using 100 mesh. The powder was dissolved in 200 milliliters of HCl and was stirred at 80 degrees Celsius with magnetic stirrer for 120 minutes. The solution was filtered and the resulting filtrate was added with methanol and stirred to form precipitated pectin. It was purified with ethanol and dried in the oven at 50 degrees Celsius. Next, the preparation of the edible film. 3 types of solution were prepared in the study. The first solution was prepared by dissolved pectin powder in 150 ml of distilled water. The second solution was of distilled water, and the solution was heated and stirred with magnetic stir. Next, all of the prepared solution were put into a glass beaker and stirred and heated. Then 1.5 milliliter of glycerol was added to the mixture solution and it was further stirred and heated to 90 degrees Celsius. The solution was poured into a mold and dried in the oven at 90 degrees Celsius for 12 hours. The mechanical properties of edible film including tensile strength, elongation, and modulus Young. The test result of tensile strength of edible film can be seen in figure 1. Test results show the edible film was made tapioca flour had tensile index of 4.27 megapascal. Edible film with tapioca flour of 0.6 gram had tensile index of 5.23 megapascal. Edible film with the addition of tapioca flour of 1.2 gram result in a tensile index of 8.22 megapascal. Moreover, with the addition of tapioca flour of 1.8 gram and 2.4 gram, tensile index were 7.62 megapascal and 1.86 megapascal. Tensile strength test showed that tensile index of edible film increased with increasing of concentration of tapioca flour until reaching optimum tensile index at tapioca flour of 1.2 grams. It showed that tensile strength value were influenced by the composition of the tapioca flour. However, tapioca flour addition of more than 1.2 gram decrease the tensile index of edible film. The test result of elongation percentage of edible film can be seen in the figure 2. Test results show that the elongation of edible film were obtained range between 40.4 to 45.7%. The addition of tapioca flour didn't affect the elongation percentage of edible film. The results show that the optimum percentage of modulus Young of edible film was reached at 1.203 megapascal with addition of tapioca flour at concentration of 1.2 grams. Percentage of modulus Young of edible film increased with increasing of concentration of tapioca flour. The conclusion. The study showed that edible film made from tanduk banana peel pectin had following mechanical properties: tensile strength, elongation, and modulus Young. Mechanical properties of edible film such as tensile strength and modulus Young was affected by the addition of tapioca flour in edible film making. The optimum mechanical properties of edible film was obtained at addition of 1.2 grams of tapioca flour. Here is the references that we use in this research. Thank you for your attention.
Thank you, Mrs. Murietti, for the comprehensive presentation. Without wasting further time, let us continue to the third presenter. The third presenter is Mrs. No Afifa from Research Center for Appropriate Technology, Indonesian Institute of Sciences. She will deliver her presentation on accelerated shelf-life testing of Mokatila chip using critical moisture content approach and models of sorption isotherms. Co-host?
Good afternoon. My name is No Arifa. I represent my team, Jeffrey and Lista. We present our study about accelerated shelf-life testing Mokatila chip using critical moisture content approach and model sorption isotherm. Background of the study: corn cassava rice are the three highest agricultural commodity in Indonesia, which have good nutrition content, and we've developed the flour mixture become Mokatila chip, which enriched with tempeh. In other said, the shelf life is the important attribute informed to customer. So food industry needs short time to determine it, and the chips are easily damaged due to absorption of water vapor. So the proper approach is the critical water content using Lavoisier equation. The aim of our study is to evaluate the moisture content substance isotherm behavior of Mokatila chip and to estimate its shelf life in PET, PP, and aluminum foil packaging using critical water content approach. Material we used in our study are modified cassava flour, cornstarch, corn flour, rice flour, baking powder, salt, cooking oil, and tempeh. Mokatila chip was made following procedural report by Afifa et al. And salts we use in this study to create some RH, sodium hydroxide, magnesium chloride, potassium iodide, sodium chloride, potassium chloride, and barium chloride. Moisture content was determined by drying the sample using open under constant actuator. And initial moisture content was measured after chip were dried immediately. Critical moisture content was measured when the chip flexibility was rejected by panelists through sensory tests. And equilibrium moisture content was determined by exposing the chip in chamber containing 6 different salts to maintain water activity in the range 0.07 to 0.9. Some substance isotherm model we use in our study are Gap, Oswin, Smith, Henderson, Halsey, and BET model. We can see the equation from equation 1 to equation. This is we can see that the higher LH, the greater equilibrium moisture content we get. After we input this water activity value to equation 1 to 6, we get moisture content prediction from some model. And from this figure, we can see that chip follow sigmoidal substance isotherm type. And from this curve, we can see that in the first segment, or low relative humidity, moisture content rise slowly. However, at her age about 67%, large amount of moisture was absorbed and the curve sharp rising. It suggests that the chip deteriorate faster above 67%. And this is the constant equation we can use Microsoft Excel. This is the CAP model. And from this table, we can see that CAP, Oswin, and Smith appropriate to describe the moisture substance isotherm phenomenon of chip, because for MLD below 5 shown that the exact substance isotherm model, and from this table we can see that CAP, Oswin, Smith have MLD less than 5. Among this model, CAP model was the most suitable model to describe the substance isotherm behavior of chip. And initial moisture content is 1.4%, and this is fulfill Indonesian national standard for exterior snack that required below 4%. Critical moisture content we get after sensory test about 5.5%, and for equilibrium moisture content after we input in CAP equation at ambient RH in Indonesia about 80% is 0.147 gram water per gram solid. To calculate safe life using Lebuscher equation, besides initial critical and equilibrium moisture content, we need other data like solid weight and packaging properties. Mokatila chip in this study packed in 25 gram weight, and after corrected with initial moisture content, we get dry weight 24.6 gram. And packaging we use in this study have like 17 meter and 10 in weight, so we get packaging area about 0.034 meter square. The package permeability is defined as the water vapor amount that can pass through a package per day at certain atmospheric condition. Based on table 3, we can see that PET packaging has the highest packaging permeability, following with PP packaging and aluminum foil packaging. The higher packaging permeability value, the higher transfer of moisture entering packaging, so that the shelf life of product become shorter. The shorter shelf life was found in product packed with PET and then PP and aluminum foil. So the conclusion of our research is accuracy test of moisture substance isotherm model was obtained by MRD 0.73 for the CAP model. The self-life Mokatila chip are stored in the PET, PP, and aluminum foil packaging was 3, 8, and 60 mon respectively. Thank you very much.
Good. Thank you, Mrs. Noafifa, for the presentation. Before we jump into the fourth presenter, let me remind you kindly that you can use the chat room to pop up question to the previous presenters. Please write your question briefly and clearly. All right, we will continue to the fourth presenter, which is Mrs. Agustin Susilawati from Research Center for Chemistry, Indonesian Institute of Sciences. Her title is Modified Cassava Flour or mocha as food additive on infant instant cream soup using fortification of natural folic acid. Co-host.
Good afternoon, ladies and gentlemen. Let we listening and look how my presentation about our research in subtitle role of mocha as food additive on infant instant cream soup using fortificant of natural folic acid. This research from Research Center for Chemistry, Indonesian Institute of Science, and I'm Agustin Susilowati, and our team in this research is Mr. Aspianto, Mrs. Yati Mariati, Mrs. Mina Artanti, Mrs. Puspa, Mrs. Hakiki Melanie, and Ms. Setiani Budiarti. How about folic acid? We know folic acid is very, very important as precursor in synthesis of nucleotides at period of healthy cell fission and growth. This is more important, especially for infant. How we can get a fortificant of natural folic acid? We mix tempeh. You can using a mung bean or soybean tempeh, and fermented vegetable. You can using broccoli and spinach. But in this research, we using a fermented broccoli and nixtamal yellow corn. I show you the photo of fortificant. We using fortificant A and B, which fortificant A is mixture of soy tempeh, nixtamal, yellow corn, and fermented broccoli, and fortificant B is fortificant mixture of mung bean tempeh, nixtamal, yellow corn, and fermented broccoli. So the different of fortificant is using different tempeh. How can we application for get instant infant cream soup? We using mocha flour as carrier of folic acid for complementary feeding for infant. You can look for fortificant this mocha flour and the basic formula of soup for get instant infant cream soup. The aim of experimentally, this is just no best mocha concentration, as carrier. We using fortificant END at mocha concentration of 0, 6, 12, 80 to 24 and 60% of base formula of infant cream soup. Analysis, using analysis will dissolve protein, total solid, total sugar, reduction sugar, folic acid. Forget of characteristic using LCMS to know how about monomer folic acid dominant, particle size distribution, and volatile compound by GCMS. How can we get instant infant cream soup, and how on application for ready to serve as for cream soup? First, we preparing fortificant natural folic acid by mix of fermented broccoli and soy of mung bean tempeh, and then mixing and adding nixtamal, yellow corn 1 part, and then homogenizing, and then drying. So we get a powder of fortificant folic acid. Forget instant powder of infant cream soup. We mix a soup formula, base soup formula, and just on drying mixing, and for to ready a chef, we can adding hot water and mix so, and then pouring and mixing, so we get a set in instant infant or cream soup research. And in this research, you can look how characteristic of material. Table 1, you can look how composition, fortificant A, B, base formula of soup and mocha, and you know how the material have a different composition, especially for folic acid. How effect of formulation on composition on infant instant cream soup. The process dry mix and about dissolved protein. We can using fortificant A on concentration of mocha of 30%. This is how about effect for total sugar, total solid and reducing sugar in different concentration mocha. A condition of optimum process for best concentration mocha, we get 30%. You can look how instant infant cream soup A, I mean using fortificant A, get a folic acid optimum. About identification for folic acid monomer, this is a chromatogram, and you look one peak T, 0.6, and on mass spectra, using look how dominated of monomer with molecule weight for 440.075 Dalton. How about distribution of particle size? We just saw how on optimum condition process. So just show you how particle size on infant instant cream soup is on average size 141.026 micron. This identification of volatile compound. It shows how instant infant cream soup is dominated with a compound of acetic acid, furan, and derivative compound, methyl ester, and its derivative compound and fatty acid. You look how why we get acetic acid. It's because we using mocha who we get from fermentation by lactic acid. In conclusion, based on folic acid, optimization treatment was achieved at mocha concentration 60% using fortificant A. In this optimization system, increase folic acid about it's so a lot. I think more than tenfold when compared with each component in fish formula without you adding mocha as food uh, as food. And then how about instant in fact giving characteristic as I said to you before at monomer folic acid and particle thank you so much for Ministry of Research, DIPI, and Indonesian Institute of Science for funding this research. Thank you so much for all attention. Please don't ask too much.
Okay, thank you so much, Mrs. Agustin, for your presentation. We will straight go to the Q&A session. I will pick 2 questions first, and then after that, we will look at the time and continue if we still have time. The first 2 questions is from Mrs. Emma Damayanti from LIPI to Mrs. Muriati from Jakarta State Polytechnic. The question is, could pectin also be extracted from other types of banana peels or other fruit peels? Why do you choose tanduk banana peel in your study? And then the second question is from Mr. Ahmad Sarifudin from LIPI to Mrs. Asri also from LIPI. The question is, what is the application of your finding for the fibrose corn rice product? So, co-host, please unmute to unmute Ibu Muriati and also Ibu Asri. Thank you.
Okay. Thank you, Mrs. Emma, for your question. Pectin could be extracted from pears, apples, plum, orange, and citrus. The fruit contains small amount of pectin. In this research, we use extracted pectin from tanduk banana peels because the previous study showed that the yield of extracted pectin from tanduk banana peels range from 16% to 24%. Thank you.
Okay, thank you for the brief answer. We will continue. Ibu Emma, have you feel satisfied with the answer from Ibu Muriati?
Yes, thank you.
Okay, then we continue to Ibu Asri.
Thank you.
I'm sorry, is that only me that cannot hear the answer? Has everybody hear the answer clearly?
Maybe you have to turn your volume a little bit higher.
Ibu Asri, can you repeat your answer with a higher volume and talk loudly?
Okay. Okay, our research. Okay, can you hear my voice?
Yes.
Okay, to predict equilibrium moisture content if this product is stored under various RH condition, and if this research is continue, it can be used to estimate the shelf life and design a storage area that is suitable for product characteristics.
State Polytechnic. In producing edible films, bubbles usually arise, which will affect the mechanical properties. So, what did you do? What are your efforts to minimize the bubbles? And then, how about the water vapor transmission rate value of the edible film? Because the ability to withstand gas transmission also affects the quality of the edible film. And then, the fourth question is from Mr. Ahmad Sarifudin again to Ibu Agustin Susilawati from LIPI. The question is, how about the smell of fermentation notes in the instant cream soup sample? Is there any leftover smell? If there is, how do you overcome this of notes? To co-host, please unmute Ibu Muriati and then Ibu Agustin. Ibu Muriati, please answer the question.
Thank you, Mrs. Nok Afifah and moderator. This research was carried out until the stage of making edible film and mechanical testing of edible film. Thank you for your suggestion, Ms. Nok Afifah. The next stage, we will testing the wafer vapor transmission rate for edible film. In this research, we get small of the bubble because the open reaction. Jadi we get small of bubble in this research. Jadi we don't do anything to minimize the bubbles. That's it. That's my question. Thank you.
So without any effort, the Bible is not so much. So that's why you don't do special effort. Okay.
Yes. Okay.
Hang on. Ibu Nur Afifah, are you satisfied with the answer from Ibu Muriati? Please unmute yourself before answering.
Thank you for your answer.
Okay, thank you. Then we will continue to Ibu Agustin, who will answer the question from Mr. Ahmad Sarifuddin. Co-host, please unmute Ibu Agustin. Co-host. Okay.
Can you hear my voice?
Yes. Yes. Please.
Okay. Yes. For Mr. Ahmad Syarifuddin, I'm very thanks for all your question. I just said to you, I'm on wondering too, because on GCMS we get acetic acid. It must not smell, but when you pouring the soup, it's nice. So in this research, it's not about pouring of soup, just on instant soup. So I understand for your question about smell. No, not smell, but I understand because on fermentation using mocaf, you can get acetic acid, and I'm surprised too, because not allowed researcher get this compound on the product, but in real it's not on smell. It's not on real, because the concentration is very small. So maybe in my research about pouring on instant infusum, later I show you how the data it's in I said to you about how smell on real product. No, I'm not for find the smell. It's nice, but in this research just on instant. So like instant cream soup usual. No smell.
Right. Hello. Okay.
Hello.
Yeah.
Hello.
Yes.
Okay.
Mrs.
Harry,
Agustin.
can you hear my voice?
Yeah. Yes. Okay. As I understand, in the GCMS, you can see a lot of things, including the acetic acid. However, in the real product, it doesn't smell like that. It just smells nice. Maybe because the concentration is low. So, Bapak Ahmad Sarifuddin, hello.
Yeah, Bu. Okay, Bu. Okay. Thanks for your answer. Yes, thank you for your answer, and I'm waiting for your next presentations. Thank you.
Yes. You can look.
Thank you so much.
How I data. Thank you so much, Mr. Sarifuddin.
For your collaboration.
Thank you, Ibu Agustin. Okay.
Waiting for the collaboration, Lee.
Okay, I will have a look if we still have further questions. Hang on, let me scroll. There isn't any more question in the chat room, so this is my turn to ask question. I have question for Ibu Muriati. I'm wondering if you would like to assess the glass transition of your edible film.
Beg pardon, please.
I'm wondering if you would like to assess the glass transition to check the melting, so that if your edible film can melt in the mouth or not.
Oh, gitu. The edible film has to dry in the oven, ya, in the mold. We use the petri dish in the mold. We dry it in the oven at 90 degrees Celsius. That's it.
So, will you have a look at the glass transition temperature or not?
Yes, we use temperature at 90 degrees Celsius with the oven, right in the oven. Gitu.
And it melt at 90 degrees Celsius?
Yes, yes.
Oh, okay. All right.
With the oven at 90 degrees Celsius for 12 hours in the oven, right in the oven.
Okay. The only presenter that hasn't get any question is Ibu Nok Afifah. Is there anybody who would like to ask Ibu Nok Afifah? Hello? If there is not anybody, I will ask question to Ibu Nok Afifah. Here I read that you also use tempe to enrich your mozzarella chips. I'm wondering if your mozzarella chips smells beany or rancid during the accelerated shelf-life testing. And how will you improve the off smells?
What smell?
Beany smell or rancid smell, because you also put tempeh in your composite flour. Do you have any off flavor or beany smells from the tempeh?
Oh no, because we just add about 10% of tempeh, and in the process before tempeh mixing with other flour, tempeh we blending
Okay.
so it will reduce smell and other enzymes to repair the final quality. Process blending before the mixing of this flour.
Okay, so you did cleansing before you turn the tempeh into flour to overcome the smells. All right. Thank you so much. So we already finished. We already finished the first part. Let me check. Okay, we will continue to the second part. In the second part, we will have 3 presenters. Let me check first if all the presenters for the second part already available. Okay, all right. All of 3 presenters already there. We will continue straight to, I'm sorry, I'm not so sure it is Mr. or Mrs. Woro Setiapuma from Research Center for Appropriate Technology, Indonesian Institute of Sciences, with the title Quality of Physicochemical and Sensory of Mushroom, Pleurotus ostreatus, chicken nuggets, with carrageenan and konjac as hydrocolloids. Co-host?
Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Let me introduce myself. My name is Woro Setiapuma from Research Center for Appropriate Technology. I and Dita Kristanti studied about nugget, and I would like to present our research. Quality of physicochemical and sensory of mushroom chicken nugget with carrageenan and konjac as hydrocolloids. Chicken nugget is one of popular frozen food that is chicken product from meat cut to shape and cut of bread flours. Today's consumer preferences are healthy food. Consumption of fiber have been important because it affects on our system body. Substitution of frozen food is one of ways to improve product being better. Mushrooms have been known as starch fiber, hence they have been used as meat-like product. They have unique flavors and beneficial effect. They have rich in protein, high fiber, low cholesterol, and rich in amino acid. In addition, the texture of mushroom is similar to chicken meat. Chicken nugget is made from meat and flours that is established butter on emulsion system. The emulsion system will impact on quality of the nugget. Additional thickening agent is one of methods to preserve the quality of nuggets. Sodium tripolyphosphate, STPP, is food additive that commonly used as a tenderlace. However, the STPP is chemical and its additional is regulated. Hydrocolloid is a food additive. This is a thickener and it will absorb water easily and form a gel. Therefore, this is important to understand split product and food additive prior to the quality of nuggets. The aim of this research was to investigate the effect of various hydrocolloids on the physicochemical characteristic of mushroom chicken nugget. And this is material and methods. The mushroom was obtained from Bukdes Jaya Maju Bersama, Subang. STPP, konjac, and carrageenan flours were obtained from Bandung. The formulation of the nugget is shown in Table 1. We had 4 formulas, NO, N1, N2, and N3. After we obtained the nugget were analyzed. The analysis were moisture content, ash content, protein content, cooking loss, texture analysis, and sensory evaluation. The data that we obtained was analysis. Using analysis was performed by ANOVA and Duncan's multiple range test at 5%. Now we will focus on result and discussion. Chemical properties of mushroom nuggets is shown in Table 2. Moisture content was significantly different. Based on the Standard National Indonesia, SNI, the moisture content of chicken nugget was less than 60%. The water content determines shelf life of product, which increasing of water content results microorganisms to multiply. According to additional of hydrocolloid, the moisture content of N2 was the best. All treatment nuggets did not affect this ash content. This result was corresponding with SNA that the ash content of chicken nugget was less than 2.5%. pH value of all nuggets was 6.10 until 6.26. The pH value of N1 and N2 were significantly higher than N0. Furthermore, the pH value of N3 was slightly higher than N0, but not significantly different. The pH value impact on water binding in meat because higher pH will increase total negative change of myofibrillar protein and the water binding effect on meat. No significant differences of additional hydrocolloid on protein value. However, the value of protein N2 was slightly higher than N3. Meat protein from shrimp stable net of restructured product like nugget, where water and denaturation of protein affected restructured product. Next is physical properties of nugget. Cooking loss of N2 was the lowest. The cooking loss is character where indicated water holding capacity. Result indicated that hydrogen bond of hydrocolloid was interacted with meat along mushroom. Higher cooking loss, the lower water and protein interaction bonus. Lightness value of N2 were not significant different than N3. The nugget commercial is made from chicken, while additional mushroom can change of the color. Previous research, the mushroom have grayish white color, then additional mushroom can decrease lightness. Redness is determined by a positive value in the A measurement of the colorimeter. Chicken nugget with high B value indicate yellowish appearance. In this research, the A value of NO, N1, and N2 did not differ but significantly higher than N3. The B value was significantly lower in N1, N2, and N3 than NO. The hardness was affected by the addition of hydrocolloids. Hardness of NO, N2, and N3 were not significantly different than N1. The change in hardness is possibly because the nugget were solid interaction with other ingredients, and the N2 had better texture profile than NO and N3. This is a sensory evaluation of this research. In this present study, no difference of flavor, texture, and taste was found, indicating that all these samples had been accepted by panelists. The color and overall of N1 was the highest. However, the color and overall of other treatment were accepted by panelists, which based on the overall was more than 3 value. Finally, we will conclusion of this research. We have 4 conclusions. The first is the moisture content and pH value were affected by additional of hydrocolloid, whereas N2 was better than N3. The additional hydrocolloid were affected the cooking loss and texture. The cooking loss, lightness, and hardness of N2 were higher than NO and N3. Sensory attributes of the mushroom chicken nugget were unaffected by additional hydrocolloid. The study describes that konjac and carrageenan can be used to replace STPP without noticeable change in sensory. The last is the additional of konjac was better physicochemical properties of mushroom chicken nugget. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you so much for the nice presentation and the nice picture of the nuggets. It looks yummy, especially if we are already hungry in this ready session. Now, we will continue with the next presenter. The next presenter is Anastasia Weni, another Anastasia from Research Division for Natural Product Technology, Indonesian Institute of Sciences. The title is The Antioxidant and Antibacterial Study of Canarium indicum L latex extract. Co-host, please help with the video.
Good afternoon, everyone. Thank you very much for the chance. Today, I would like to present our research about the antioxidant and antibacterial study of Canarium indicum L latex extract. My name is Anastasia Weni Indrianingsih from Research Division for Natural Product Technology, Indonesian Institute of Sciences, LIPI BPTPA LIPI, Yogyakarta. As we know, the free radicals can come from our surrounding environment, such as from sunlight, from alcohols, from fast food, from pollution, tobacco, and chemicals. It can also come from physiological and biological process in our body. It would lead to oxidative damage to human body, for example, lipids, protein DNA, and also can cause several chronic diseases, such as atherosclerosis, cancer, diabetes, and aging. Several studies has revealed that the intake of natural antioxidants has been associated with reduced risk of cancer, cardiovascular disease, and diabetes. And plants may contain a wide variety of free radical scavenging molecules, such as phenolic compounds, nitrogen compounds, vitamin, terpenoid, that rich in antioxidant activity. Natural compounds isolated from plants as rich sources of novel drugs have become interest of many research. Meanwhile, the genus of Canarium has approximately 100 species worldwide. Canarium indicum L, or canary in Indonesian language, is one of the species of Canarium. It is an indigenous plant in Indonesia, especially in the eastern region such as Maluku Island. The distribution of C. indicum plants spread in various countries, including Indonesia, the Philippines, Papua New Guinea, and Malaysia. The seed of C. indicum is usually used as a snack food. The oil from C. indicum seed also comparable to other plants, such as cashew nut, hazelnut, and macadamia nut. Moreover, the other parts of C. indicum, such as leaves, stems, and exocarp of C. indicum seed also had been studied for treating disease and natural food coloring. However, the bioactivity studies about latex of C. indicum is still very rare. Because of that, the aim of this study is to determine the antioxidant and antibacterial activity of C. indicum latex extract. For the methods is as follows. So the preparation of C. indicum extract, the powder of C. indicum latex was macerated using ethanol solvent for 2 days at room temperature. It was then filtered and evaporated using vacuum rotary evaporator to obtain ethanolic extract of C. indicum latex. And then the FTIR spectroscopy analysis was conducted by mixing the samples with KPR to make KPR pellet. The spectra were recorded in the range frequency of 4,000 to 500 per centimeter. The DPPS radical scavenging assay was conducted according to Yen and Chen, 1995. So several concentration of samples was dissolved in methanol. It was then reacted with DPPS 1 millimolar concentration and incubated at dark room temperature for 30 minutes, and the final solution was measured at 570 nanometers using ELISA multi-plate reader. The antibacterial assay was conducted according to Wulanjati et al. 2020. The method was agar-well diffusion, so the extract was dissolved in dimethyl sulfoxide at several concentration, and then 100 microliter of bacteria suspension was inoculated in nutrient agar plates. The extract solution was then added into the wells in the solid media. After incubation at 37 degrees Celsius for 24 hours, the diameter of inhibition unit was measured. This is the yields of Canaricum indicum latex extract shown in the tables. So the yields is around 44%. The latex of the indicum was rarely studied. However, the nut of the indicum were extensively studied. Several literatures reported the composition of C. indicum nut and kernel. Carbohydrate, vitamin E, and fatty acid, including palmitic acid, stearic acid, oleic acid, and linoleic acid, were obtained from the kernel of C. indicum. This is the antioxidant activity of C. indicum latex extract using the DPPH assay. As we can see here from the figure, that based on the DPPH assay, the higher the concentration of C. indicum latex extract, the higher the radical scavenging activity. The C. indicum latex extract has high activity as of around 10.9 at 100 ppm, 12.7 at 200 ppm, 27.1 at 400 ppm, and 88.4% at 800 ppm. While the IC50 of the C. indicum latex extract is around 697.4 ppm. Meanwhile, the antioxidant activity of the ascorbic acid standard is around 76.7 at 25 ppm. A literature, Jaskarsi et al. At 2017 reported that the C. indicum nut contains phenolic and flavonoid compounds, so that it possesses a high antioxidant activity. The quantification of phenolic substance in C. indicum methanolic extract is of 7.4 to 8.8 milligram per gram of gallic acid equivalent. This is the antibacterial activity of C. indicum latex extract. So we can see here that for the concentration extract of 500 to 1000 ppm, but for bacteria E. coli and S. aureus, there is no inhibition unit diameter was observed. However, at 10,000 ppm, for the E. coli, it was observed around 2.4 millimeter. Meanwhile, the inhibition zone for S. aureus at 10,000 ppm is around 6.4 millimeter. Meanwhile, the ampicillin as the positive control show the inhibition zone around 3.1 millimeter. From these results, we can assume that the C. indicum latex extract show moderate antibacterial activity against E. coli and S. aureus. This is show the FTIR spectra of C. indicum. As we can see here that the several functional group vibration was observed for C. indicum latex. For example, in the absorption peak around 3425.6, it is a broad peak. It possibly indicated the functional group vibration of OH from phenol or alcohols. Meanwhile, the sharp peaks around 2H62 to 2924 indicated the functional group vibration of CH alkane. Meanwhile, the sharp peak around 1635.6, an indication as a functional group vibration of CC aromatics, and in 1705.1 sharp peak It possibly indication the functional group of CO from aldehyde, ketone, ester, or carboxylic acid. From this study, we can see that the FTIR spectra of C. indicum show that it possibly contains phenolic compounds. This is for the conclusions that C. indicum exhibit a potent antioxidant and antibacterial activity, and it's also show moderate antibacterial activity against E. coli and Castor aureus. And it is likely that the antioxidant and antibacterial activity was attributed to the phenolic compound of C. indicum latex extract. However, more functional use and bioactivity of the C. indicum latex extract still need to be further to be performed. That's all of my presentation today. Thank you very much for your kind attention.
Thank you, Ibu Weni, for the presentation. Before we continue to the last presentation, I kindly remind you to pop up question in the chat room. Please write your question briefly and clearly. Now, we shall continue to the last presentation. The last presentation will be delivered by Mr. Ahmad Sarifuddin from Research Center for Appropriate Technology, Indonesian Institute of Sciences. His title is Cold Water Swelling Starch, Method to Prepare and Recent Application. Co-host, please help with the video.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. In this precious moment, I would like to share a review and my research result related to the preparation and recent applications of cold water swelling starch. I will start this presentation from the requirement to modify native starch due to its limited characteristic to satisfy in certain applications. Generally, there are 4 methods to modify starch. From those modification by physical and enzymatical methods do not produce dangerous byproduct. Therefore, they are classified as a green method. Cold water swelling starch was firstly developed to overcome the low solubility of native starch. It absorbs water instantaneously, giving a cook-like texture. Therefore, it is used as a major ingredient of ready-to-consume products, such as instant porridge and puddings. There are several terminology of cold water swelling starch, such as cold water soluble starch, granular cold water swelling starch, or ethanol treated starch. There are many methods to prepare cold water swelling starch. One of the most efficient and we can consider as a green method is ethanol heating method, in which starch is heated in ethanol solution. Let's take a look at the main characteristic of cold water swelling starch, which is solubility. Similar to the other starch product, the solubility of cold water swelling starch increase in basic or acid solution. It also increases at elevated temperature. Cold water swelling starch produced from higher temperature tended to have higher absorption and solubility. It is well known that the structure of native starch are converted during cold water swelling starch conversion. The compact native crystal of starch, either O and B, is destroyed upon conversion. Instead, V-type crystal is formed. This crystal is an artifact of complex between single helix of amylose and ethanol, which is located within the helix. In our recent investigation, we observe there are 6 stages of starch conversion process. Let's take a look at the morphology of cold water swelling starch. When the cold water swelling starch is in a dried state, It looks like a string pillow with a corrugated surface. Once it absorbs water, it swells immediately, holding the water inside its structure. In our latest investigation, we also employed nitrogen solution isotope to observe the pore characteristic of cold water swelling starch. We observed that cold water swelling starch contain narrow, non-rigid slit shape randomly connected. Consequently of these properties is that it will swell immediately once in contact with water. The specific surface of cold water swelling starch also increase from the cold water starch produced from the higher temperature. We also observe the pore characteristic distributions of cold water swelling starch. Most of the cold water swelling starch, the pore are within the size mesopore and macropore. It means that the size is greater than 10 nanometer. In our latest investigations, let's take a look at the application of cold water swelling starch. The cold water swelling starch are commonly used as ingredients of ready-to-consume products such as jelly, porridge, and gel. It also can be used for encapsulating agents such as for dyes, drugs, and nutraceutical. In our latest investigation, we employed the cold water swelling starch as a binding agent of medicinal tablet. We observed that the hydration properties of medicinal tablets prepared from cold water swelling starch show different hydration properties. The granular ETS, it will disperse immediately, while tablet from non-granular ETS, it will still intact. Because different hydration properties, it will also affect the release properties of the active ingredients encapsulated by ETS tablet. We observe, or we can conclude that tablets prepared from cold water swelling starch are suitable for control release system. That's the end of our presentation. Thank you for your attention. Any comment, question, and further collaborations are expected. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Thank you, Mr. Ahmad Tarifuddin. So that makes the last presentation. Now we can continue with the Q&A. As before, first. The first one is from Bu Novita Indrianti to Ibu Woro. The question is, when you analyze the physical properties of the meat mushroom nugget, in particular the cooking loss and the texture profile, did you use nugget with coated or uncoated with breadcrumbs? The second question is from Mr. Ahmad Sarifuddin to Mrs. Weni. The question is, in what product will you apply your latex canarium? And then, if you already done it, what is the activity after application? So, I would like to ask the co-host to unmute Ibu Woro and Ibu Weni. Ibu Woro, the time is yours.
Thank you very much for Ms. Indriati for the question. In our research, we use coated nugget in GPA analysis. Is it clear, Mrs. Indriati?
I'm sorry, I cannot hear. Can everybody hear the answer from
I don't hear your voice, Miss Moro.
Ibu Woro.
Okay, I will try to
repeat your answer clearly. Please be close to the microphone.
Can you hear my voice?
A bit louder, please.
Okay. Okay. In our research, we use coated nugget in TPA analysis.
Coated nugget, it means with the breadcrumb, yeah?
Yes.
All right, Ibu Novita Indrianti.
Okay, but I think it's better if you analyze the profile texture of the profile and the physical properties of nugget if you don't uncoated with breadcrumb. So the result is not biased.
Biased by the breadcrumb. Okay.
Yes.
So that's a suggestion from Ibu Novita to also complete the study, the coated and the uncoated. Okay.
Thank you very much for your suggestion. Maybe we will use the uncoated nugget in our next research to avoid the bias.
Thank you so much. And then the next one is Ibu Weni will answer the question from Mr. Ahmad Sarifuddin. Ibu Weni, please.
Thank you very much, Mr. Ahmad Sarifuddin, for your questions. Actually, we conduct this research because we are very curious about the biological activity of the latex of C. indicum, because usually many research only conducted on the nut or on the kernel of the C. indicum. So, however, for further research, I think we need to conduct the isolations of active compound, so we can know precisely what active compound that contain in the latex extract, because usually the latex extract of the indicum now is only used as varnish or on the resin application. So we hope in the next research, if we can isolate the active compound, maybe we can apply it to some food supplement or nutraceutical, but not from the isolation, but from the synthesis of the new active compound, if it is possible. I think this is my answer. Thank you. And from Ms. Emma Damayanti, is it okay if I answer that questions?
Hang on, I will ask Mr. Ahmad first. Mr. Ahmad Sarifuddin, are you satisfied with the question given by Ibu Weni?
Yeah, I just wondering because the latex is not common to be used in a food product. So what Bu Weni has explained that is maybe applied for furniture, but I mean on the future, it's possible maybe because maybe it's not appropriate for food, but if this still has activity, it's possible to be applied in a cosmetic product, maybe this is my opinion. Thank you.
Okay, so that's just a suggestion from Mr. Ahmad Sarifuddin to Ibu Weni. Maybe you can consider the application in cosmetics. And then we will continue.
Thank you very much, Mr. Ahmad Sarifuddin, for your suggestion.
The third question apparently also meant for Ibu Weni. This question is from Ibu Emma Damayanti to Ibu Weni. Please explain that the basis of an extract was. Please explain how the basis of an extract was claimed to be active, moderate, or inactive as an antioxidant. And then what is the concentration level for each criteria?
Okay, thank you.
Please.
Thank you very much for Ms. Emma Damayanti for the questions. Actually, usually in natural product isolation, below 50 or 100 ppm is included in an active extract, and between 100 to around 300 is moderate, and up then more than 300 or 500 ppm is considered as not active. However, recently I found some other journal, in food journal, they said that around IC50 around 1000 ppm is still okay. So in here, I think that it was our choice to choose which one is appropriate in our research. I think that's my answer.
All right. Ibu Emma Damayanti, are you satisfied with the explanation given by Ibu Weni?
Okay, because in my opinion,
please speak louder, Ibu Emma.
Hello?
Please speak louder.
Okay, okay. As far as I know, the natural product is considered active, maybe below under 100, 400 for IC50, and then active and potential under 10 IC50. Maybe it's a different reference and maybe in different application for a natural product. Okay, but it's okay if Ibu Weni the extract from canary latex claim is moderate activity, maybe because it's crude extract. Is right?
Yes, crude extract, yes.
Okay, okay. Okay, I think enough. Thank you.
Okay.
Thank you, Miss Emma.
Both Ibu Emma and Ibu Weni. There isn't any more questions, so I will ask question. My first question is for Mr. Ahmad Sarifuddin. Your presentation about cold weather swelling starts. Can you give us some example what have your office done with the development of cold water swelling starts? Hello.
Thanks for your the questions, Bu Anas. Actually, this is part of my dissertation. So this is in this year. Actually, we don't do anything with this project. So maybe in the future. So I hope that I can find any collaboration from this meeting. Thank you.
Oh, okay. So not yet done anything with this. All right. My further question is for Ibu Worosia Buma about the mushroom nugget. I'm wondering if you already compare the chicken nugget with and without the addition of carrageenan and cognac hydrocolloid, is there any difference in oil absorption after the frying? Co-host, please unmute Ibu Woro. Hello, Ibu Woro.
Can you hear my voice?
Yes, I can hear your voice. Have you heard my question?
Can you repeat again, please?
Okay. When you did your study, did you compare between the nugget with the additional carrageenan and cognac and without the additional? Is there any difference after frying about the oil absorption? Like the nugget?
Okay, actually, we don't did assay after
trying, but we did a cooking loss of this treatment, and it can I show my presentation?
Yes. Thomas, please help Ibu Woro to share, to allow Ibu Woro to share screen.
Yes, please.
Okay, in my research, the cooking loss of NO is higher than additional hydrocolloid. It indicated that in NO, there is release properties in nugget. Then the treatment N2 and N3. And it means that additional hydrocolloid it's good enough in nugget to keep the oil absorption. Is this clear?
What do you mean to keep the oil absorption? To keep avoiding or to keep gaining?
To avoiding.
To avoid, okay.
Yes.
All right. Thank you for your explanation.
Yes, thank you very much.
I already checked the chat room, then there is a new question. This is from Ibu Nok Afifa to Bapak Ahmad Sarifuddin. What are the advantages of cold water swelling starch compared to other starch modification methods? And actually, what important properties do you want to get from this modification?
Okay, thank you for the questions, Bruno. Because cold water swelling starch, the main purpose of why we should modify the native starch and convert it to cold water swelling starch is that we want to get the instant properties of the flour or the starch. So most of applications is that they apply this cold water swelling starch as ingredients for a ready to consume product. So that in terms of properties, of course, we have to take a look at the properties of instant flour, which is the main properties is the solubility, of course. Yes, that's the way my answer.
Ibu Nok Afifa, are you satisfied with the answer from? Okay, thank you, Mr. Ahmad, and your little assistant and your background. Okay, I still have one question for Ibu Anastasia Weni. Hello, Ibu Weni. I would like to know why did you choose ethanol as your solvent? Because as far as I know, the solubility is for latex in ethanol is not so good. But please explain. Maybe you have other reason.
Okay. Thank you very much, Miss Anastasia, for the questions. So usually for natural product, the universal solvent we use is methanol or ethanol because that both of solvents can extract many kind of compounds. However, in this case, I use the ethanol because it would be more food grade or more safe compared to methanol. And in this research, the latex extract form is already solid, so we powder it and then dissolve in macerate in ethanol. I think that's my answer.
All right, thank you. So it's because of you're talking about the safety of the final product, so that's why you choose ethanol.
Yes.
All right.
Thank you.
Okay. Is there any other question for the presenters? We still have about 9 minutes. Hello. While we are waiting for the question, if any, I would like to remind you that we will have a short break at 3 p.m. But then at 10 minutes past 3 p.m.
Because it's liquid, it's more easier to dissolve it with the acid. We can use AS, or we can use ICP-MS for more sensitive analytical methods. I think that's my answer, Mr. Safaat.
All right, thank you, Ms. Enda Damastuti. I hope that will answering your question, Mr. Safaat. Okay, I think this will be the very last question of our session today. So I would like to close this session. So as we finally come to the end of this session, I'd like to thank all of the speakers for a very informative and interesting presentations, and also to all of the audience for the active participations. Thank you so much. We hope that you enjoyed today's session. And just a quick reminder that we will be having a closing remarks at 16:45 at the main room. It will be greatly appreciated if you can join our closing ceremony. So thank you for your attention and see you at the closing ceremony. Thank you, everyone.
vision begins with a spark of inspiration. And each dream is driven by the passion to realize it. For over 50 years, we have worked hard to serve Indonesia. Becoming a world-class research institution that uses research to improve the nation's competitiveness is our vision at the Indonesian Institute of Sciences, or LIPI. LIPI was formally established in 1967, but its history goes back as far as 1817, with the establishment of a plant conservation center at the Bogor Botanical Garden. We are spread over 11 provinces. We have over 4,800 employees. We are the largest research institute and the largest patent contributor in Indonesia. LIPI is directly responsible to the President of the Republic of Indonesia, with the coordination of the Ministry of Research, Technology, and Higher Education. LIPI's acting chairman is Professor Dr. Bambang Subianto, who is assisted by the Vice Chairman, General Secretary, and 5 deputies who each oversee research centers and technical service units in their respective fields. LIPI has a strong foundation built on conducting research that delivers large-scale, significant, and real results for Indonesia. LIPI's research focus areas include life sciences, consisting of molecular biology, biotechnology and medicine, biomedical science, molecular and post-genomic agriculture, exploration and utilization of biodiversity research, earth sciences, consisting of marine biology and inland waterways, eco-hydrology, coastal and marine areas, climate change and disaster management, and mineral resources research. Engineering sciences, consisting of defense and IT security, telecommunications and electronics, advanced materials and nanotechnology research, social sciences and humanities, studies into scientific and technological developments and scientific community training. LIPI researchers are supported by world-class facilities spread over 48 research centers and technical implementation units throughout Indonesia. LIPI also has research vessels for oceanography research activities, as well as other marine research expeditions. We understand the importance of maintaining the natural ecosystem and its contents. To that end, LIPI has collections of flora and fauna from all over Indonesia collected and recultivated in its 5 botanical gardens: Bogor Botanical Garden, Cibodas Botanical Garden, Purwodadi Botanical Gardens, Bali Botanical Gardens, and Cibinong Botanical Garden. LIPI manages world-class flora-fauna collections. The Bogorienza Herbarium Museum, established in 1817, is the 3rd largest herbarium museum in the world. The museum has the 10th largest reference collection of fauna specimens in the world and and Indonesia Culture Collection, or INA-CC, which holds LIPI's collection of microorganisms listed in the World Microorganism Collection Culture Federation. Given its vast resources, LIPI is also appointed as a scientific authority in 1. Guiding, training, assessing, and authorizing national researchers. 2. Accrediting scientific journals. 3. Studying and evaluating biodiversity. Establishing botanical gardens, as well as scientific testing and measurement. LIPI also plays a strategic role in various international scientific activities. This is evidenced by LIPI occupying the trusted position as the international focal point for UNESCO and has 3 international centers, namely Asia Pacific Centers for Eco-Hydrology, APCE, International Center for Interdisciplinary and Advanced Research, ICR, and Marine Biodiversity and Ecosystem Health Center, or MARBEST Center, engaged in marine training. In addition, LIPI is also active in other international organizations, such as Science Council of Asia, SCA, Botanic Gardens Conservation International, BGCI, International Council for Science, ICSU, and others. LIPI works with the following stakeholders to disseminate its research results to the community, namely, national ministries and state-owned enterprises in the field of metrology and testing, local government, universities, and industries, including the private sector. Since 2010, more than 171 MOUs of cooperation have been established between LIPI and the national stakeholders. This cooperation has resulted in the establishment of various science and techno parks. These parks incubate technology startups and facilitate technology transfer. LIPI has also established more than 70 partnerships with international partners since 2010 in various branches of science across the world. Including research collaboration, workshops, and exchange programs and scholarships for LIPI researchers. For more than 50 years, LIPI has produced research that has significantly improved the quality of life of Indonesians, as well as the development of science in Indonesia. To encourage the spirit of research among the younger generation, LIPI has, since 1968, hosted research paper competitions for teenagers, as well as the National Young Inventor Award, which is keenly followed by students throughout Indonesia. We are ready to cooperate with various elements, both national and international, to realize our vision of becoming a world-class research institution to improve the competitiveness of Indonesia in the future.
Yogyakarta, the city of education and culture. But then, how can we fulfill our longing for these traditional foods when we are far away? Research and Development Division for Natural Materials Technology, Indonesian Institute of Sciences, has been developing packaging technology for ready-to-eat foods. Traditional foods are processed and packaged in cylindrical can, so it has longer period of shelf life and increased its value. The implementation of packaging technology developed consistently in the institution. The institution has been working along with partners to implement the packaging technology. They manage to process and package such as pindang egg, slices of chicken meat, krecek condiment, tolo beans, tofu, and tempe. This traditional food became an example of our canned traditional foods. Let us show you the process. First, cans are sterilized in autoclave. An autoclave used high-pressured, high-temperature steam and of the technology is able to enhance the production capacity of canned foods and increase the sales turnover in Yogyakarta's traditional foods processing industries. Furthermore, these culinary industries are getting more knowledge on the hygiene, safety, and standardization of food processing technology. The packaging technology has received a certification of Good Manufacturing Process or GMP from the Indonesian National Agency of Drug and Food Control. In addition, canned traditional foods have received positive responses from many people. Yogyakarta will always be the city that feels like home. However, it's always been nice to be back to Yogyakarta, the city of yearning with the greatly delicious traditional local food.
ISAC, Indonesian Science Expo, is the one of the biggest scientific festival in
Good afternoon, ladies and gentlemen. We really appreciate for your participation until closing ceremony. Our honorable speakers, moderators, participants. Finally, we come to closing ceremony of the ISAC 2020. We ask the Director of Research Center for Chemistry to closing remarks. Dr. Reni Meliana, the time is yours.
Thank you, Marisa. Assalamualaikum and good afternoon, ladies and gentlemen. Alhamdulillah, I believe that during these 2 days, all of us have obtained a fruitful discussion in this 6th ISAC 2020, which main theme is natural medicine and nutraceutical. We have learned a lot from the keynote speaker delivered by our keynote speaker, Professor Gunawan Indrayanto, thank you. Dr. Ambara Rahmat Pradipta, Dr. Muhammad Iqbal Qauderi, we meet again. Niles Nirmal, PhD, also Associate Professor Nurul Huda and Professor Dr. Nina Artanti. Once again, on behalf of Research Center for Chemistry, LIPI, and the committee, thank you so much for delivering such important material so that all of us can deepen our knowledge. Furthermore, I would like to thank to all presenters and participants who have joined this conference. I certain that every presenter have presented their own research. I sincerely hope that through your presentation and discussion afterwards, all of us can contribute for better research in the future, and we can create good collaboration among us. Finally, I would like thanks to all the committee members for all this hard work organizing this conference and ensuring that it processed smoothly. I would like also to apologize for any inconvenience that you might experience during this conference. Ladies and gentlemen, all the participants and honorable speakers, once again, I thank you for your participation and wish you all stay healthy. See you again in the next 7 ISAC 2021. Thank you, Marisa.
Thank you, Dr. Yeni, for the officially closing our symposium. Ladies and gentlemen, and all plenary speakers, it's better for us to open our video. Maybe the committee will take the last documentation for today. Okay, thank you for the open the video. Also, we would like to say thank you for spending the time with us, for all participants and also plenary speakers. And also, at last but not least, we hope we can meet on the 7th International Symposium of Applied Chemistry 2021. I'm Marisa Angelina. As a master ceremony of the symposium, would like to say apologize for the inconvenience during our symposium. Good afternoon, have a nice day, and see you. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you all.
Bye. Thank you. Nice to meet you, Dr. Niles. Hope we meet again sometime in the future.